To outline today's event, this public meeting in its entirety will run approximately 3.5 hours, which includes a Q&A session. And if you have questions now or during the meeting, please enter those questions into the Q&A chat pod. We'll answer as many questions as time allows. For opening remarks, we welcome Dr. Teresa Mullins, who's the CEDAR Associate Center Director for Strategic Initiatives. She oversees areas of strategic interest to the center and external stakeholders, leading a variety of CEDAR efforts, including patient-focused drug development. She also leads CEDAR's international program, leads the FDA delegation to ICH, where she serves as chair of the ICH Management Committee, and represents FDA on the IPRP Management Committee. Please join me to welcome Dr. Mullins.
Thank you, Ray, and good afternoon, everyone. So I'm not going to reintroduce myself. Thank you very much for that nice introduction, Ray. And I'll just begin with saying that ICH has a mission of harmonization for better health, and this has been true since it began in 1990. And today, this mission seems more important than ever as we contend with climate change, the COVID pandemic, regional political, military conflicts, and other things that have created disruptions to supply chains and operations and to drug development. There is just this growing need for rapid, efficient, and continued development and delivery of drugs to patients who need them, both to address emerging disease threats and growing prevalence of chronic disease around the world, and the disability and premature death and suffering caused by that. Now, since 1990, ICH has been operating, but only after the reforms of 2015 did we really expand this organization to include a really diverse and wide membership. So it began with only 6 members representing regulators and industry and 4 observers, but today we have 20 members and 35 observers at ICH, and that are contributing to the work of 32 working groups that involve over 1,000 people, including over 700 experts on the working groups, over 300 people involved in supporting these groups. And so it's become an incredibly large and productive endeavor to produce modern tools and approaches to drug development, and including clinical studies and modernizing manufacturing approaches. The range of topics is very impressive, and so is the extraordinary scientific leadership of the experts that you're going to hear from today. And so we are offering you, FDA and our Health Canada colleagues are together offering you a very rich program, I think, today, starting with an overview from Nick Orfanos, and then covering a wide range of topics. So bioanalytical method validation, electronic structured harmonized clinical study protocols, drug interaction studies, bioequivalence for immediate release solid oral dosage forms, selective data safety collection for late stage or post-approval studies, rodent carcinogenicity studies, the assessment and control of DNA reactive mutagenic impurities, biodistribution studies of gene therapy products, viral safety evaluation of biotechnology products, continuous manufacturing. It's just an incredibly vast list, and all of these areas are really vital to continue to develop and deliver medicines, safe and effective and high quality medicines to the patients who need them. And so we're very pleased that you could join us today, and I want to thank you again for that, and look forward to the discussions and presentations. And with that, I'd like to turn it over to Nick to provide you with the overview of ICH. Over to Nick.
Thank you very much for those opening remarks and for the introduction. Good afternoon, everyone, and good morning to those tuning in from the West Coast. As mentioned, my name is Nick Orfanos, and I am the ICH Coordinator for Health Canada, and I will be opening today's presentations with an overview of ICH. For those who may be unfamiliar with the ICH, I'll be providing a brief overview of the organization, its approach, and its process for harmonizing international drug regulation standards. ICH was established more than 3 decades ago by representatives of the regulatory agencies and industry associations from the US, Europe, and Japan. Health Canada, Swissmedic, and the WHO also provided valuable contributions from the beginning. ICH seeks to foster the development of uniform, scientifically driven standards to improve the efficiency of new drug development and registration processes, and to promote public health so as to prevent duplication of clinical trials in humans, and to minimize the use of animal testing without compromising safety and effectiveness. These objectives are achieved by developing harmonized technical guidelines and standards implemented by regulatory authorities around the world. In 2015, ICH made a series of significant organizational changes to extend the reach of harmonization beyond its founding regions. It became a nonprofit legal entity under Swiss law. The reforms were designed to benefit patients around the world by focusing global pharmaceutical regulatory harmonization work in a single forum for constructive dialogue on scientific issues. Also by promoting more involvement from international regulators and global industry sectors. Also by facilitating greater adoption of new and improved research and development approaches, common standards and therapeutic advances, and finally by providing a more stable and efficient operating structure. As mentioned, a driving factor of its organizational changes was to make ICH a truly global initiative and enable an expanded forum for dialogue on scientific issues. ICH has grown to include 20 regulatory and industry association members spanning over 4 continents. Also 36 observers, including regulatory authorities, regional harmonization initiatives, international industry pharmaceutical organizations, and other organizations affected by ICH guidelines. Last year, the MHRA UK became ICH's newest regulatory member, and DPM from Tunisia became its newest regulatory observer. ICH is governed by the ICH Assembly, the overarching body, including all ICH members and observers that approves new topics for harmonization, adoption, amendment, or withdrawal of ICH guidelines, admission of new members and observers, and the election of Assembly and Management Committee leaders. The Management Committee is a smaller body, including permanent and standing members of ICH and elected members that oversee the operations, including administrative and financial matters and oversight of working group efforts. It recommends to the Assembly the selection of new topics for harmonization and adoption, withdrawal, or revision of guidelines, and it includes a financial committee that develops ICH budgets and allocates funding for ICH operations. This slide provides a picture of the ICH governance structure. At the center, you'll see the ICH Assembly, the ICH and MEDRA Management Committees branch to each side. The Secretariat provides day-to-day management of ICH, including preparing and documenting the ICH Assembly and Management Committee meetings. ICH coordinators are selected by ICH members and facilitate the efficient operation of ICH harmonization activities. They serve as the central point of contact for their organizations with the ICH Secretariat and facilitate conversations between the ICH Management Committee and working groups. Over its history, ICH working groups have produced and updated approximately 70 harmonized technical guidelines on all aspects of human drug development. ICH has also pioneered development of electronic standards for the transfer of regulatory information, creating an electronic common technical document that brings together information in a harmonized format accepted by regulators in all ICH regions. The electronic common technical document has dramatically reduced the burden on developers seeking marketing approval for a drug product in multiple regions by allowing them to submit data and information organized in the same way to each regulator. ICH has also developed a medical dictionary for regulatory activities terminology, otherwise known as MEDRA, to facilitate sharing of regulatory information internationally. It has become a global standard in the protection of public health. MEDRA is a rich and highly specific standardized medical terminology used for registration, documentation, and safety monitoring of medical products both before and after market entry. Since its launch in 1999, MEDRA has grown steadily. It is now available in 14 languages, has over 6,800 subscribers, including regulatory authorities, pharmaceutical companies, clinical research organizations, and healthcare professionals in over 125 countries. ICH also develops questions and answers documents and training materials to support the implementation of ICH guidelines, as well as reflection papers to provide a more strategic approach of new topic uptake, which I'll be touching on in upcoming slides. This slide provides a sample of harmonized topics considered by ICH working groups. As you can see, the topics range from safety, efficacy, quality, to multidisciplinary efforts, including electronic standards. Multidisciplinary efforts can also include guidelines that cross-cut across different disciplines, such as quality and safety. The 70 ICH guidelines previously mentioned cover these and other topics and are based on a wealth of foundational knowledge and experience provided by experts participating on ICH working groups. Last year, over 1,000 individuals were contributing to the work of ICH, either as representatives or delegates of ICH bodies, expert working groups, or in other supporting roles. As you can see from this chart, over 700 technical experts participated on 34 ICH working groups last year. More than a third of these experts come from recent ICH members and observers, reflecting a significant level of interest and engagement by ICH's newest parties. The harmonization of ICH topics begins with an annual new topic selection process. ICH members and observers submit proposals for new topic harmonization on an annual basis. A subcommittee of the management committee evaluates and prioritizes the proposals and presents recommendations to the ICH Assembly for final decision to move forward with the development of a new ICH guideline or the revision of an existing guideline. Following ICH Assembly endorsement of a new topic, the ICH Assembly endorses the concept paper outline, which is developed by the proposing party. An informal working group is then established to develop a final concept paper and business plan. The concept paper is meant to outline the scope of the guideline, while the business plan provides an outline of the timing of when the guideline will be finalized. The management committee approves the final concept paper and business plan, and a formal expert working group or implementation working group is established. Following the establishment of the expert working group, drafting of the guideline begins. At step 1, the expert working group develops a consensus draft of the guideline. When the draft is completed, the topic leads for each party represented on the working group sign off on the guideline at step 1. At step 2, first the assembly endorses the draft guideline. Second, regulators separately do so. Step 3, the guideline then moves on to a public consultation phase in which it's submitted for public comment in a transparent process designed to solicit thoughts from a wide array of stakeholders. Members solicit public comments from their regions, and then the draft guideline is also posted on the ICH website for others to offer comments. The expert working group collects all of the comments, reviews and evaluates them, and revises the draft guideline into a final guideline. At step 3, the working group's topic leaders sign off on the final guideline. At step 4, the ICH Assembly and its regulatory members approve the final guideline. And finally, at step 5, ICH regulatory members and observers implement the final ICH guideline within their respective country or region, achieving the goals of harmonization. The status of implementation efforts is published on the ICH website and updated periodically by the ICH Secretariat. As part of its effort to enhance international acceptance of harmonized standards, ICH develops training materials based on the scientific and regulatory principles outlined in the guidelines. These include a training library on the ICH website, including training materials and Step 4 working group presentations, funding support for training programs organized by ICH members and observers, ICH recognized training programs hosted by a variety of organizations, associations, regulatory authorities, and academia. These are offered in person, virtually, and online, and information is provided on the ICH website. Other online training materials include some translations. So following this presentation, you hear updates from experts participating in ICH working groups that have recently reached ICH milestones, either at Step 2 or Step 4. So as a recap, Step 2 guidelines that have recently been released for consultation in ICH regions, and Step 4 recently finalized guidelines. Just as a brief update, we would like to outline the new ICH topics that have either recently been launched or will be launching soon. So launched in spring of 2022 was targeted revisions and additional issues in the Q1, Q5C series on stability testing. A new guideline on model-informed drug development, another which was launched as well in the spring of 2022. A new guideline on pregnant and breastfeeding individuals in clinical trials, just very recently launched in January of 2023. And two new guidelines that are awaiting some resources to free up in the quality space are Q6, the revision to Q6, which is a new quality revision on specifications, as well as a new topic which we'll be looking at as a standard on the structured product quality submission. So to wrap things up, ICH is a unique organization that draws on the expertise of regulators and industry to achieve international harmonization of technical guidelines, ultimately to benefit patients. It is used as a transparent, science and consensus-based process for guideline development that includes opportunities for public comment, and it includes commitment of regulators to implement the guidelines. ICH has extended global participation and engagement through recent reforms. And I hope this introduction has been helpful. Thank you very much for your time and attention and interest in our efforts.
Thank you for that introduction to ICH. We'll now move to updates on ICH efficacy-related guidelines. The first presentation, M10 Bioanalytical Method Validation and Study Sample Analysis, will be presented by Dr. Anna Edmondson, Senior Clinical Assessment Officer with the Pharmaceutical Drugs Directorate, Health Canada. The M11 Clinical Electronic Structured Harmonized Protocol will be provided by Vivian Combs, Advisor, Process Owner, Clinical Systems and Supply Planning, Eli Lilly and Company. Then M12 Drug Interaction Studies will be presented by Dr. Raj Matabushi, Associate Director for Guidance and Scientific Policy in the Office of Clinical Pharmacology, Center for Drug Evaluation and Research, also known as CDER, FDA. The M13A Bioequivalence for Immediate Release Solid Oral Dosage Forms will be presented by Dr. John Gordon, Senior Clinical Assessment Officer with the Pharmaceutical Drugs Directorate, Health Canada. And finally, the E19, a selective approach to safety data collection in specific late-stage pre-approval or post-approval clinical trials presentation will be provided by Dr. Mary Tanhai, who is Deputy Director, Clinical, in the Office of New Drugs, CDER FDA. Please join me in welcoming our first presenter, Dr. Edmondson.
Thank you for the introduction. It sounds like we're going to have a lot of interesting presentations today. My name is Anna Edmondson, and I work in the Division of Biopharmaceutics Evaluation at Health Canada. I will start off the talks in this session with the ICH M10 guideline on bioanalytical method validation and study sample analysis. I will begin my talk with some background on the guideline. The main focus of my talk will be changes from the Step 2 draft document. These would be changes, wordsmithing, and clarifications that came about after discussion of the comments that we received in public consultation. Finally, I will speak of updates since sign-off of the finalized guideline. The M10 guideline provides recommendations both for the validation of bioanalytical methods and the analysis of study samples, and the guideline was renamed as shown in red at the top of the slide to more accurately reflect its purpose. M10 ensures the quality and consistency of bioanalytical data, and this includes the analysis of both chemical and biological drugs in biological samples, and it harmonizes current regional guidelines, which will facilitate drug development. M10 was developed in October of 2016 based on a concept paper and business plan. The draft Step 2 document was published for public consultation on February 26, 2019. And as a result of the public consultation, we received about 2,500 comments. These comments were triaged and were either incorporated into the guideline, flagged for a Q&A document, or as potentially requiring training, and I'll speak more on that later. M10 was signed off as a Step 4 document on May 24, 2022, and could then be implemented by the ICH regulatory members. This is a high-level overview of the content of the M10 guideline. As stated in Section 1.3, the scope of M10 is studies submitted to make decisions on and/or support the approval, safety, efficacy, and labeling of a drug product, and this could include pivotal toxicokinetic and pharmacokinetic non-clinical studies and all phases of clinical studies. I'll start with updates to the chromatography section of the guideline, first validation and then study sample analysis. Wording was added to Section 3.2.5, Accuracy and Precision During Validation, to provide recommendations for non-accuracy and precision runs. They are that the low, medium, and high QCs should be analyzed in duplicate, and that at least 2/3 of the total QCs and 50% per concentration level should be within 15% of their nominal values. In Section 3.2.7, Dilution Integrity, the terminology was changed from dilution ratio to dilution factor for consistency. Additionally, the wording was updated to state that the dilution factors and concentrations applied during study sample analysis should be within the range of the dilution factors and concentrations evaluated during validation. Section 3.2.8, Stability, had a few updates in order to provide clarity. The requirement for 3 stability samples was reworded. It was clarified that one bulk QC should be prepared at each concentration level. For each concentration tested, the bulk sample should be divided into a minimum of 3 aliquots that will be stored, stressed, and analyzed. The section on multiple analyte stability was also changed for clarity. There was confusion regarding the wording of specific drug regimen, and it is now clarified that this section applies to fixed dose combination products and specifically labeled drug regimens. So it only applies when 2 compounds are labeled to be administered together, such as ritonavir administered as a booster with an HIV drug. The types of stability tests that this is applicable to was included, and the terminology was changed from QCs spiked with all analytes to QCs spiked with all of the dosed compounds. And this is important because an analyte is something that's measured, and the requirement to measure these dosed compounds is outside the scope of this guidance. And what's important is that all dosed compounds be included in the QCs for stability. The section outlining the recommended stability tests was reorganized. The content has not changed. So stability of the analyte in matrix, stability in process samples, stability in stock and working solutions, and finally, stability of the analyte in whole blood should be evaluated. 2 further updates of note are that for the stability of the analyte in process samples, it's stated that the total time that a process sample is stored must be concurrent. So you cannot add autosampler and other storage times together. The study sample requires 10 hours of autosampler stability. It can't be taken from 5 hours in the autosampler and 5 hours of a process sample stored in the refrigerator. For the stability of the analyte in whole blood, it's been clarified that this should be conducted when the matrix used is plasma. The requirement for serum has been removed. For Section 3.2.9, which is reinjection reproducibility, the purpose of the assessment has been clarified, which is to establish the viability of process samples and support their storage prior to reinjection. Additionally, suggested criteria for establishing reinjection reproducibility have been included, and that is to reinject a whole run that would be comprised of calibration standards and at least 5 replicates of the low and high QCs after storage. And the precision and accuracy of these reinjected QCs would establish the viability of these process samples. Moving on to study sample analysis for chromatography. a few modifications were made to section 3.3.2, acceptance criteria. Wording has been added regarding the case that multiple batches are included in an analytical run to state that calibration standards in a failed batch cannot be used to support the acceptance of other batches within the analytical run. And in runs containing diluted samples, there was previously a requirement that the dilution QCs be diluted using the same dilution factor as the study samples being diluted. A sentence has been added for the case that multiple dilution factors are used in one analytical run, and it now states that only the lowest and highest dilution factor needs to be included. During study sample analysis, when several analytes are being assayed simultaneously, a valid result for one analyte should not be rejected if the other analyte fails. And for comparative bioavailability, bioequivalence, or BABE studies, a separate table reporting values from rejected runs is required. Now I will discuss changes from the Step 2 document for ligand binding assays, or LBAs. First, validation, and then study sample analysis. In section 4.2.2, selectivity, it's stated that blank samples should be obtained from at least 10 individual sources. In the final guidance, a caveat has been added that the use of fewer sources may be acceptable for rare matrices. This section of the draft guideline had a suggestion to assess selectivity in relevant patient populations, and in the final guideline, examples of these populations has been provided as shown here. In section 4.2.4, accuracy and precision for the validation of LBAs, similar wording to that in the chromatography section has been added for non-accuracy and precision validation runs. And in this case, the acceptance criteria are, again, 2/3 of the total QCs, and at least 50% per concentration level, but within 20% of nominal values as it's for LBAs. Section 4.2.7, stability, had a few updates regarding QC preparation in order to provide clarity. The requirement for 3 stability samples was reworded in a similar manner to the chromatography section, and it was clarified that 1 bulk QC should be prepared at each concentration level, which would then be split into 3 aliquots. It's acknowledged that there may be a need to freeze macromolecules overnight, and it now clarifies that QCs should be frozen for at least 12 hours between thawing cycles. Multiple dosed compound stability wording has been added. This wasn't present in the draft guidance, and it's the same wording that we see in 3.2.8 for chromatography, but again, a caveat is added for LBAs that this should be conducted or assessed on a case-by-case basis. Moving on to study sample analysis for LBAs, these reflect changes that were made in the chromatography section. In 4.3.2, the statement that calibration standards in a failed batch cannot be used to support the acceptance of other batches within the analytical run. And for the reanalysis of study samples, the requirement of a separate table reporting values for rejected runs for BABE studies only. I will speak about changes in the remaining sections of the guideline. In Section 5, incurred sample reanalysis, it has been clarified that the acceptance criteria are to be within plus or minus 20% for chromatography and within plus or minus 30% for LBAs. Section 6.1, partial validation, there was an addition for consistency. In the LBA methods section, it now states that a change in anticoagulant in biological fluids would be a possible reason to conduct a partial validation. Section 6.2 is for cross-validation, and this section has been reworded slightly for clarity. Cross-validation is required to demonstrate how reported data are related when there are multiple bioanalytical methods or multiple bioanalytical laboratories involved. And it should be noted that if your data are from different fully validated methods and the data are not to be combined across studies, cross-validation is generally not required. Section 7.1 for endogenous compounds has been updated, mainly wordsmithing to clarify that this section is for methods for analytes that are also endogenous molecules. And Section 7.2 on parallelism for LBA methods has been reworded, essentially explaining that this is a study-specific issue that is assessed within a study rather than during validation, along with minor changes to the wording of the assessment criteria. There were minor modifications to Section 8.1, summary information, predominantly terminology changes. In the draft guideline, a requirement for a list of regulatory site inspections for BABE studies was included. In the final guideline, it specified that included inspections should be from inspections conducted over the 3 years prior to the study and 1 year post-study completion. There were minor modifications to Section 8.2, documentation. In the draft guideline, requirements specific to BABE studies were embedded within the overall requirements in each section, and in the final guideline, the specific requirements for BABE studies have been separated out, so they're much clearer now. For chromatograms and reintegration, for non-BABE studies, it previously stated that randomly selected chromatograms from 5% of studies submitted in the dossier should be submitted. It's now clarified that it should be randomly selected chromatograms from 5% of samples submitted in the dossier. And the requirement for a separate table reporting values for rejected runs in BABE studies has now been included in the documentation table. Since sign-off of the guideline in May 2022, the accompanying frequently asked questions, or FAQ, document that was published on May 25th, 2022 was subsequently split into 2 documents. There's a new Q&A document, and it contains the majority of the content from the original M10 FAQ. There is also a new FAQ document, and this has the leftovers, the 3 remaining FAQs that were not put into the Q&A document. And these 3 remaining FAQs, the intent is that they will be incorporated into the training slides, and then this new FAQ document will also be removed from the ICH website. The FDA posted the M10 guideline to their website on November 2022, and Health Canada in January 2023. Training slides are being finalized by the expert working group, and these are examples to illustrate certain aspects of the guideline. When the public consultation questions were being triaged, questions that had a more complex answer were moved to training, and this to-be-published set of training slides should provide clarity on the thinking behind some parts of the document. In summary, the finalized M10 guideline will result in the same scientific regulatory requirements being applied in different regions. It avoids unnecessary duplicative testing and supports streamlined global drug development. Thank you for listening today, and thank you to the members of the ICH M10 Working Group, especially Dr. Brian Booth, my counterpart from the FDA, who was our regulatory chair. And I will be joining the panel at the end of the webinar for the live Q&A. Thank you.
Thank you for that introduction. We'll move past the obligatory notice. Today, I'll be covering the value proposition of the ICH M11 protocol template. I'll provide an orientation to the materials that have been delivered, the protocol template, the guideline, and the technical specification. We'll take a closer look at the technical specification, and then I'll wrap up and be part of the Q&A at the end. So the reason why ICH formed an expert working group was really related to the fact that there's no internationally harmonized standard template, either for format or content of protocols. And so that makes things very inefficient and inconsistent. Specifically, it can lead to quality issues, such as delaying timelines for drug development. It's very resource intensive to fix those quality issues. It's inefficient. We're oftentimes duplicating effort, and we're not able to use the information to its full value, nor can we exchange it efficiently. So there's a lot of opportunity to make this better. The value that M11 brings is it does establish a common place for things, a common table of contents. It highlights the basic requirements for protocols. I think the impact on the whole clinical development space will be somewhat similar to what we felt with CTD and ECTD. There's a lot of parallel concepts at play. And it's a foundational step toward a digitized, which I think has been dreamed about for many years. It makes the whole experience more predictable for those who are reviewing protocols. Predictable structure, predictable content. You can anticipate the same level of detail and same style of presentation in some areas. We'll all be working toward common instructions, aiming at a common target. We still will have some flexibility with needed, but we can approach the work with these commonalities and the assurance that it's consistent, that what the protocol template is calling for is consistent with what's required by other ICH guidelines and will be acceptable in all of the ICH countries. You may be wondering, why did we need to have an electronic template? What's wrong with just a Word document? Nothing's wrong. But, you know, I'm old enough to remember when submissions used to look like the picture on the left, both submissions of protocols as well as new marketing authorization. We took a giant step forward about 20 years ago with electronic submissions. So now we submit PDFs. But I'm not sure from a utility perspective, we're certainly killing fewer trees, but I'm not sure that the information in that PDF is really any more useful to us than the paper was. It's really just electronic paper. And what we really want to do with M11 is unlock the value of the content, enable us to do more sophisticated things with the information inside the protocol, not just moving around at a document level. So the electronic aspect starts to granularize the content so that it can be exchanged and moved around, and we can apply more sophisticated technology tools. It sets the stage for additional standards development for certain parts of the protocol that could use even more standardization and technological enhancement. And it creates sets the stage for the development of informatics and software development as well. So on the left are kind of the benefits of just any template, Word or otherwise, that come from having that common format and common structure. But then with the electronic aspects, we get more searchable content and metadata. You can search on indication or population, who the sponsor was, what drug class we were using, amendment history and changes. You can create a tailored experience. The view of the information might look different depending on a reviewing tool. The experience of a site might look different than what a reviewer would be looking at. Still based off all the same protocol content, but different aspects of the content could be surfaced according to the needs of who's looking for it. It enables collaboration and continuity, much easier during mergers and acquisitions to fold an asset into a development program or move it out or have joint sponsorship of studies across companies. And it, as I've alluded to, sets the standard or sets the stage for future standardization. So moving to a little bit of orientation to M11 itself. This is a kind of a bird's eye view of the ICH step process. So it's a consensus-based process, starting with the expert working group at the bottom, working up to the management committee. Then there's public consultation and some revision. And then After adoption by ICH, it goes back out to the regulators for codification in local regulation. So M11 is actually at step 3. We are out for public consultation now, looking forward to adjudicating all comments and getting to step 4, hopefully by the end of the year. Because many of the existing ICH guidelines provide guidance on things that have to be manifested in the protocol, it's very important that our template be aligned with existing guidance and emerging guidance. And so that was especially challenging in developing this template, to make sure that we were fully aligned with many, many other guidelines. This is a small listing, not exhaustive. That was an extra challenge that we had. In terms of what we've delivered, there is a guideline. Like any other ICH guideline, ours is fairly high level. Associated with the guideline are 2 other pieces. There is a template, which is a Word document similar to any Microsoft Word template you may have ever seen. And there's a technical specification, which starts getting at how do we exchange information in an open and non-proprietary way, and move that protocol information around in a more granular fashion than just a whole document level PDF. So the way that we think about these is really, I think about it like a glass of water. So the guideline is sort of the container. It's, as I said, pretty high level, not expected to change a whole lot over time, while the template and the tech spec are like ice and water. So in the same way that, that ice and water are different forms of the same matter, so too the template and the tech spec. So these are versioned. They're expected to change over time with science and technology. But they restate the same details. So we're probably more familiar with the template, but the technical specification really articulates the same things, the same principles in a different way, technology and more machine understanding. The objective, certainly, the purpose of the guideline is to describe our general protocol design principles that we followed when developing the template. And then we wanted to make sure that what we delivered would be acceptable to all of the regulatory authorities of the ICH region. So that's the big objective. In terms of scope, the templates that we've delivered, template and tech spec, are really scoped to interventional clinical trials of drugs, vaccines, and drug-device combination products when they are registered as a drug. It's appropriate for all phases of research and for all therapeutic areas. A few things that are out of scope. The guideline and the template and tech spec are intended. They are not intended to specify processes related to the development and maintenance of a protocol. That's we are silent on those topics. That's generally the purview of the sponsor. They also don't supersede or negate any other guidelines that establish requirements for protocol content. So that's one of the reasons why making sure we were aligned with existing guidance was important. And we don't provide instructions that are going to help you design a great study or do a fantastic job of crafting a protocol. This is really about just the basics of what information goes where within a protocol document. Taking a closer look at the technical specification, again, this is the other way of articulating what's already in the template. So the tech spec presents the business requirements and the protocol content components that are articulated in the template. It'll also provide the rules around conformance and cardinality and the other technical attributes that enable electronic exchange of the content, many of which are also inherently expressed in the template. We don't think about it in those terms, but a lot of those are articulated indirectly within the template as we know it. And just to hit this point again, the technical specification will be an open, non-proprietary standard. We're not going to be tying anyone to a particular technology or a particular solution system kind of situation. So to fully develop the technical specification requires modeling of the content. So that means there are aspects that are modeled at the whole document level, and then each section is modeled with headings and variables, and then there are components within each section that are further modeled and may contain variables, pick lists, headings, fields for a running narrative. All of that's being modeled out so that we can exchange it electronically and apply electronic tools to it. So for each one of those components that I said we were modeling, they're on the prior slide, if you open our technical specification, every component gets a box like this. And in that box, we're describing what's supposed to be in that component. Is it a specific field? Is it a block of content? What are the other defining attributes? And what are the business rules that need to apply? So a lot of this is populated based on what's in the protocol template, and it will continue to mature. I mentioned that in a lot of places where we allude to the cardinality and the conformance requirements in the template, but we have to formalize that in the technical specification. And part of that formalization will come through engagement with a standards development organization. So we did publish the technical specification now. It's not fully mature. There are some blank spaces in it. That's by design. And that part of it will continue to mature. The template itself is pretty mature, as you would expect for a Step 3 document. But there's more work to do on the tech spec, and there will be additional public opportunities for public comment on it. So you may be wondering, why did we release the tech spec now if we know it's not done? We wanted to give insight into our direction of travel, and specifically to get some engagement with the communities that are likely to be very, very interested in what we're doing. So software developers, IT professionals, data standards folks, data managers, and statisticians. They're the probably the most able to kind of see what we're doing and anticipate where we're headed and have thoughts about what would be good and bad about it. And so we wanted to give an early opportunity to see what we're doing. And again, we'll have additional opportunities to engage as the tech spec matures. The current version is a restatement of what's already articulated in the protocol, and it will continue to be refined as we partner with the standards development body. To wrap up, hopefully I've convinced you that a harmonized protocol template and technical specification that really help us do more granular electronic exchange can enhance the ability of sponsors and regulators, investigators, really everyone involved in clinical development to review and execute clinical research more efficiently, which ultimately helps us deliver medicine to patients faster, which is, I think, the reason why we're all involved in the we are. For more information, you can find us on the ich.org website. I've put the link here. It's easy to remember. It's ich.org. And then if you go to the multidisciplinary topics, the M topics, and search for M11, you'll find us. We are out for public comment, but the window for public comment is closing very quickly. Just within the next week, several countries are closing their public comment cycle. So there's still time to get in just under the wire, but then you can certainly review what we're doing. And as we progress, this is the website where that will be displayed for everyone. That concludes my presentation. I'll be joining the panel at the end of the webinar for the question and answer session. Thank you so much.
Greetings, everyone. My name is Raj Madabushi, and on behalf of the ICH M12 Drug Interaction Studies Expert Working Group, I'll provide an update. I will start off with a brief background on the formation and evolution of M12, followed by highlights of the draft guidelines that was published last year, and close out with current activities pertaining to public consultation and next steps. Drug-drug interactions can occur when patients take more than one drug, and this may result in either increased toxicity or decreased effectiveness, resulting in an altered benefit risk. Therefore, it's essential to evaluate the drug interaction potential for new medicinal products during the drug development. However, it's impractical to evaluate every drug interactions during drug development. Hence, systematic risk-based strategies are essential to characterize the drug interaction potential. Regulatory agencies have developed region-specific guidances to assist drug developers. For example, FDA published two guidances in 2017, one addressing in vitro evaluation and the other one for clinical drug interaction studies. The EMA had guidelines for investigating drug interactions since 2012, and a concept paper on revision was published in 2017. The PMDA published the guideline on drug interactions in 2018. Across all these region-specific guidelines, some differences exist, resulting in heterogeneous expectations, non-harmonious interpretation and translation, potentially leading to increased drug development cost, delayed patient access, and heterogeneous recommendations as they pertain to translation of the findings of these studies to labeling. This led to the formation of ICH M12 in 2019 that brought together members from 9 regulatory agencies and 6 industry organizations. The working group diligently worked to develop and draft the guideline, which was published in May of last year. And currently, we are in the process of resolving the public consultation comments and hope to finalize the draft guideline in first quarter of next year. Real quickly, let us look at the highlights of the draft guideline. The objective of the M12 was to develop recommendations that promoted a consistent approach in designing, conducting, and interpreting in vitro studies and clinical drug-drug interaction studies during the development of a therapeutic product. The aim was to cover pharmacokinetic interactions with a focus on enzyme and transporter-mediated interactions pertaining to small molecules and biologics where such interactions may be anticipated, a focus on metabolite-mediated interactions, and lastly, model-based data evaluation and prediction of drug-drug interaction using either mechanistic static models or physiologic-based pharmacokinetic modeling. The guideline has 8 major sections, and it can be broadly viewed as 4 topic areas, starting with introduction, in vitro evaluation, clinical evaluation having several topics under clinical evaluation, and lastly, appendices and references providing supporting information across all the topic areas. Over the next 4 slides, we will take a look at the highlights of each of these 4 major sections. Starting with introduction. The highlights of introduction include the concept of risk-based stepwise approach, which involves elucidating mechanisms followed by confirmation via clinical evaluation. It also touches upon the importance of prior information that may come from either in vitro evaluation or clinical mass balance studies to inform additional clinical evaluations and support labeling instructions of use. The third aspect where the introduction focuses is on the timing and the importance of early evaluation such that any unnecessary restrictions can be avoided as it pertains to concomitant medications later in the clinical development. And lastly, it articulates the scope of this particular guidance, wherein clearly metabolism and transporter-mediated drug-drug interactions are the primary focus, out of scope of this document are pharmacodynamic interactions and other type of PK interactions which may be triggered due to changes in gastric pH, formation of complexes, chelates, effect of food, and others. The in vitro section is broadly broken down into two topics. First, metabolism-based and then transporter-based. And common across these are phenotyping studies, inhibition or induction of these pathways, and the importance of metabolite pertaining to the interactions. Of note, the aspects around UGTs is a newer topic that is discussed in this harmonized guidance. Also included in the in vitro evaluation is the data analysis and interpretation as it pertains to metabolism-based interactions, touches upon all the conventional topic areas such as reversible inhibition, intestinal inhibition, and time-dependent inhibition under the subtopic of inhibition, as well as the common approaches for evaluating induction using in vitro approaches. Similarly, for transporter-based in vitro evaluations, these include data analysis as they pertain to efflux transporters, specifically PGP or BCRP, hepatic uptake transporters such as OATP1B1 or 1B3, and renal transporters such as OAT1R3, OCT2, and MATES. The new aspect that is discussed under the in vitro section are protein binding considerations as they pertain to highly bound drugs. The guideline focuses on several important areas under clinical topics, starting with a description of the various types of clinical studies that can be considered for clinical evaluation. This is followed by various considerations for study designing and planning for CYP evaluations, UGT-mediated evaluations, and transporter studies. An emerging area of interest that is described in the guideline is the potential utility of endogenous markers as it pertains to transporter evaluation. Other clinical topics include pharmacogenetic considerations, specifically how pharmacogenetic information can complement drug-drug interaction evaluation, and considerations for therapeutic proteins. There is a specific section articulating various expectations for reporting and interpretation of the results of the clinical evaluations. This focuses on data analysis considerations, the importance of no effect boundaries, and various considerations for extrapolation of the findings to unstudied scenarios. Rounding out the clinical topics is a section on risk assessment and management, wherein general considerations are provided that promote harmonious interpretation and translation of the findings to inform labeling. Lastly, some key highlights of appendices include experimental details for various in vitro studies, a description of predictive modeling approaches, both static mechanistic and dynamic mechanistic modeling approaches such as physiologic-based PK modeling, potential applications such as when such predictive modeling approaches can be used to characterize the potential for DDIs, when can they be used to recommend whether a clinical DDI study is needed, and what might be the situations where such approaches can be used to generate clinical recommendations in the absence of a clinical DDI studies. We also describe some best practice considerations when applying such approaches as they pertain to characterizing drug-drug interactions. The last part of the appendices includes illustrative lists of drugs that can be used in vitro and clinical DDI studies for CYPs, UGTs, and transporters. Those were the highlights of the draft guideline. We'll now switch gears to share with you what's currently happening. Since we published in May of last year, the public commenting period lasted close to 6 months across all the various regions, and we got comments from 25 different stakeholders, mostly from pharmaceutical companies, from 4 consortia or associations, and 1 academic commenter. All in all, we got approximately 1,000 comments, and across different commenters, there were some similar concepts that were raised. At a high level, there were 84 comments in the introduction. Some of the key areas focused there were pertaining to the timing of DDI evaluations as well as around the scope of the guideline. The in vitro section, one of the largest sections, had comments primarily pertaining to seeking more clarification around experimentally measured protein binding to inform the in vitro evaluation, as well as clarification on the in vitro to in vivo extrapolation criteria. The clinical topics included comments predominantly around the utility and expanded description of endogenous biomarkers, as well as seeking clarity around index substrates or perpetrators for non-CYPs and transporters. And lastly, the appendices had comments focused around predictive modeling, as well as clarification around some of the drugs included in the drug list. Currently, we have set up various subgroups to evaluate these comments and address them. And as part of next steps, we'll be meeting in Geneva next month to go over these public consultation comments and start addressing them. We will have a number of opportunities over this year to be able to address these comments and hope to finalize by the first quarter of next year. Before I conclude, I would like to take a moment to acknowledge the contributions of all the members of the ICH M12 working group, and thank you for your attention. That concludes my presentation. I will be back for the question and answer panel later and will now pass it over to Dr. John Gordon.
Good day, everyone. My name is John Gordon. I'm from the Pharmaceutical Drugs Directorate at Health Canada, and I'd like to continue with the ICH update process by talking about ICH M13, which is bioequivalence for immediate release solid oral dosage forms. During my talk, I'd like to cover some of the following. I'd like to talk about the objectives, scope, and organization for the M13. I'd like to take a couple minutes to talk about the current status of the M13 process. Having done that, I'd like to then talk about the first document coming out of the M13 process, which is the M13A guideline. So I'd like to talk about the organization of the M13A guideline, and then briefly talk about some of the highlights of the M13A document. And following that, I'll conclude and talk very briefly about some next steps. The overall objective of the M13A process is to provide recommendations on conducting BE studies during both the product development phase for a product and also during the post-approval phases of a product's life cycle. The products that we are specifically talking about in the M13A are immediate release solid oral dosage forms designed to deliver drugs to the systemic circulation. So M13 is looking at conducting BE studies for IR solid oral dosage forms, for example, IR tablets, IR capsules, and IR granules or powders for oral suspension. Most of the ICH regions already have BE guidelines or bioequivalence guidelines and fairly well-used guidelines at that. So the ultimate objective of the M13 process is to create a harmonized document that brings everything together, a harmonized document that can be used by the multiple jurisdictions. And hopefully this will lead to the reduction in the need to conduct multiple bioequivalence studies for multiple jurisdictions, because the jurisdictions will have similar design requirements and data analysis requirements and so on. So this will reduce that need for additional BE studies. And ultimately it's hoped that this will support streamlined global drug development. The M13 organization is a little bit complex because it consists of 3 levels or 3 tiers. The first of these tiers, the M13A, which is the first guideline in the series, is basically the foundational guideline for all bioequivalence guidelines that will follow in the ICH process, because it deals with the scientific and technical aspects of designing a bioequivalence study and analyzing the data coming out of a bioequivalence study. Now, I think it's worth noting, however, that the regulatory decisions that may be made by a jurisdiction based on the assessment of a BE study, it does not fall within the scope of the M13 process. The M13 is dealing completely with the scientific and technical aspects of designing, conducting, and analyzing a BE study. The other thing that should probably be mentioned, of course, is you're reducing the number of BE studies that may need to be conducted by harmonizing BE guidelines. That is, so similar design requirements and assessment requirements will be in place for multiple jurisdictions. So hopefully that will reduce the number of BE studies that need to be conducted. The other thing that would also reduce the number of BE studies that would be needed would be acceptance of comparator products across regulatory jurisdictions. This, of course, would again reduce the need for BE studies. However, I think it's important to note that the acceptance of a comparator product within a regulatory jurisdiction is often dependent on the local laws for that jurisdiction. And because of that, that sort of discussion falls outside the scope of M13. Again, M13 is dealing completely with scientific and technical aspects of BE study design and analysis. The second part of the M13 process is the M13B, or the second guideline in the series. This guideline is going to deal with requirements for additional strengths of a product line. So, for example, if you're developing a product that has 3 strengths, say 50, 100, and 150 milligram strengths, you're going to conduct bioequivalent studies on at least one of those strengths, that what I'll refer to as the reference strength, most commonly the highest strength in the series. What M13B then looks at is the relationship between that reference strength and the other strengths in the series, the 50 and 100 milligram strengths in my example, and what would be required in order to justify a waiver from the requirement to also conduct bioequivalent studies with those additional strengths. So M13B talks about biowaivers for additional strengths of a product line. M13C we'll talk about, it's the third guideline in the series, and we'll talk more about BE study design and analysis, but in this case it's building on the M13A process to talk about BE study design and analysis for more complex situations, such as BE studies to be conducted for highly variable drugs, BE studies to be conducted with drugs with a narrow therapeutic index, and also BE studies that with more complicated designs that sometimes come up in special situations. Now, if we could look at the current status of the M13 process. M13A, the first guideline coming out of the series, has been drafted by the expert working group and has also been endorsed by the ICH in December of 2022. Currently, this draft guideline is being posted by ICH regulatory members for public consultation. Within our region, the FDA has posted the M13A guideline for public comment with a deadline for submission of comments to the FDA set for April 3rd, 2023. For Health Canada, we're expecting the document to be posted for comment anytime now and anticipating a deadline for comment submission of sometime in late May of 2023. With respect to the M13B guideline, consensus building has already begun for the development of this guideline. It started at the last face-to-face meeting, which was held in Incheon in November of 2022, and the consensus building process has continued since then via virtual meetings and will continue on into the future. M13C has not been started yet. It's pending progress on M13A and M13B. So once the expert working group has taken M13A through all the public comments that may be received and made any revisions necessary as it moves towards step 4, and M13B progresses to a point where the EWG can then consider M13C, that work on M13C will begin. Just talk very briefly about the organization of the M13A guideline, the first guideline in the series. Of course, it's got a very standard format in that it has an introduction that talks about the objective, background, and scope. But the heart of the guideline is section 2 of the document, where it talks about the design principles for pharmacokinetic endpoint bioequivalence studies. So those are the classic bioequivalence studies that are making use of pharmacokinetic parameters such as area under the concentration time curve and maximum concentration to evaluate the performance of two products or two or more products in comparison to each other. This section 2 then goes on to talk about the data analysis for these types of studies, and it's specifically talking about data analysis for non-replicate study designs. So studies where each product is administered to a given subject only within one period and not in multiple periods in the study. So the basic classic BE study is what's discussed in this M13A guideline. The guideline does go on to talk about specific topics as well that are relevant to BE studies. For example, discussion of endogenous compounds in BE studies. Talks a little bit about how other IR dosage forms may be used, or how the guideline may be used for other IR dosage forms. Talks briefly about fixed dose combinations where you have more than one drug substance in the product that's being developed. And it also talks about pH dependency and the impact of that on bioequivalence assessment. So in just a slight bit more detail, but again in brief, the guideline talks about study population, normal healthy subjects, and talks about study design, dealing primarily with the classic BE study, which is a single dose, 2-period, 2-sequence crossover study. But it does discuss beyond that. It talks about situations where, for example, for tolerability or safety reasons, it's not possible to conduct a single dose crossover bioequivalence study in normal healthy subjects. In those cases, single dose or multiple dose studies in patients may be necessary, and that's discussed. And also it discusses the situation where you have drugs with a very long elimination half-life and where you may want to use a parallel design study as opposed to a crossover study. The study design section also talks about sample size calculation, so determining how many subjects you need to have in order to conduct a successful BE study. With respect to the products used in the BE study, the guideline does discuss the test product, and to sum it all up, essentially it's looking for the product used in the BE study to be representative of the product that is proposed to be marketed. But there are some details, of course, about the batch that's used in the study that are discussed in the guideline. Talks about comparator products, but basically the discussion is limited to identifying the comparator product as a product that's accepted by the regulatory authorities that can then be compared against the test product in the BE study. So again, it's identifying the product that the regulatory authorities will recognize as the appropriate comparator for your product and making use of that product in your BE study. In situations like I mentioned earlier, where you might have multiple strengths in your product line, you have the guideline gives you direction on which of those strengths should be used in the BE study or studies that you're going to be conducting. And of course, essentially that determination is dependent on the pharmacokinetics of the drug in question. One other study design element that I'd like to spend an extra minute talking about is the fasting or fed situation, because a lot of the elements in the study design have been harmonized across the regional guidelines. It is perhaps a little less so for the issue of whether the study will be conducted under fasting or fed conditions, or whether both fasting and fed bioequivalence studies are necessary. So this is discussed in the guideline, and basically the guideline points out that the selection of the type of BE studies, fasting or fed or both, and the type of meal that would be used depends on 3 elements. The first element, of course, being the dosing instruction for the comparator product. How is the comparator product labeled to be used? That will determine the types of BE studies that should be conducted for a product that's being developed to be bioequivalent to that comparator product. The properties of the drug substance come into question in terms of design, what bio studies should be conducted, BE studies. And finally, the properties of the products that are being compared need to be considered. And here I'm using term high-risk product or non-high-risk product, and I'm going to define what I'm referring to, or what the guideline is referring to there right now. So a high-risk product, according to the M13A guideline, is a product where the complexity of the formulation design or the manufacturing process leads to an increased likelihood that in vivo performance will be impacted differently by varying gastrointestinal conditions between the fasted and fed states. So these complex products are referred to as high-risk products in the M13A guideline. And examples of this type of products are products such as solid dispersions, microemulsions, lipid-based formulations, and so on. So these products are considered to be more complex and therefore considered to be higher risk. Jumping back to non-high-risk products and the fasted and fed situation, for non-high-risk products, the M13 guideline indicates that a fasting study would be an appropriate BE study to be conducted where the product labeling indicates that the product should only be taken under fasting conditions, or if it can be taken without regard to fasting or fed conditions. So if it can be taken fasted, should be taken fasted, or either fasted or fed, then the M13A guideline recommends that a fasting BE study be conducted for a non-high-risk product. Similarly, where the comparative product labeling indicates that the product should be taken under fed conditions for a pharmacokinetic reason, for example, enhanced absorption when administered with food. In situations such as that, the M13A guideline recommends that the bioequivalence study under fed conditions should be conducted to compare the performance of the proposed product with the comparative product. Where the comparative product labeling indicates that the product should be taken under fed conditions for reasons of tolerability, as opposed to pharmacokinetic reasons, in situations such as this, the guideline indicates that a fasting or fed study could be conducted to demonstrate the bioequivalence of the proposed product to the comparative product. Now, coming back to high-risk products and fasting and fed conditions, according to the M13A guideline, for high-risk products, BE studies should be conducted under both fasting and fed conditions for these products due to the complexity of these products. So moving along through the contents of the M13A guideline, there is a good section on the data analysis for BE studies. And again, I don't have time to go into it, but essentially it's talking about the typical PK parameters that you'll see for a BE study, such as area under the concentration time curve and Cmax, and making use of those parameters for assessment of the bioequivalence data. The guideline talks about the statistical approaches that should be used, but essentially it's recommending standard statistical approaches that are seen today to be employed for the analysis of the data. And it also outlines a bioequivalence criteria for these types of bioequivalence studies. And essentially for the primary PK parameters in your study, the bioequivalence criteria is that the 90% confidence interval for the geometric mean ratio should lie within a range of 80.00 to 125.00. And that's the bioequivalence acceptance criteria that would be applied to the primary PK parameters for the study in question. The guideline does also go on in the data analysis section to talk about studies where multiple comparator products may be used. In these situations, the comparator products are typically considered to be independent and region-specific, so the data analysis for these types of studies is still fairly straightforward. The guideline talks about situations where multiple test products might be included as well. This is a little bit more complicated, and the application of multiplicity correction in the statistical analysis depends on the underlying objectives of the study. But this is addressed in brief, at least in the guideline, to benefit those developing studies where they want to use more than one test product in the study. As I mentioned earlier, that the guideline does have a special topic section where it talks about endogenous compounds and bioequivalent studies for endogenous compounds. It provides some information for other IR dosage forms that may be being developed and how the M13A guideline can be used for those products, such as orally disintegrating tablets, chewable tablets, and there's a discussion about oral suspensions in there as well. The guideline talks briefly about fixed dose combination products, so products where you have more than one drug substance in your product that's under development and you want to compare that to comparator products. That's discussed. And finally, there's a discussion of pH dependency and situations where absorption of drug substances with pH dependent solubility that may be influenced by the gastric pH This section talks about how the bioequivalence assessment is impacted by these situations. So in conclusion, the Harmonized M13A guideline provides recommendations on BE study design for the classic bioequivalent studies, the principles for conducting these studies, and analysis of these studies, and also provides BE standards for IR solid oral dosage forms. So it's hoped that this harmonized guideline will reduce the need for additional in vivo BE studies, for different jurisdictions, and therefore support the streamlining of global drug development. And then the last thing I can say is, of course, the harmonization process will continue with the progress of M13B and M13C, and of course the completion of M13A following the comment period. Thank you very much for your attention.
Good afternoon. I'm delighted to be given this opportunity to speak to you about ICH E19 guideline, whose full title is A Selective Approach to Safety Data Collection in Specific Late-Stage Pre-Approval or Post-Approval Clinical Trials. For the remainder of my presentation, I will refer to it as E19. Over the course of the next 20 minutes, I will cover the following. Why E19 was developed, its objectives, and how it evolved over the course of 5 years. The scope of E19, the purpose of safety monitoring in clinical trials. What factors contribute to a conclusion that a drug's safety profile is well established? What do we mean when we say selective safety data collection? I will provide some examples from the guideline where E19 can be implemented and methods of implementation. Finally, I want to sum up my presentation and next steps we can take to further realize this important work. The principles of E19 were first presented in a final FDA guidance from 2016 titled Determining the Extent of Safety Data Collection Needed in Late-Stage Pre-Market and Post-Approval Clinical Investigations. So why do we need an ICH guideline? First, there was not wide knowledge of this guidance, and if known, it was not widely accepted or implemented for a variety of reasons. One of the key reasons was that drug development is a global endeavor. A protocol introducing selective safety data collection in one region but not accepted in another complicated the operations of the trial and may lead to risk that results from the global program would not be accepted by some regulatory authorities for marketing authorization. It became evident that alignment among regulators and industry was essential to advance principles laid out in this previously published guidance by FDA. The objectives of E19 are to provide recommendations regarding appropriate use of a selective approach to safety data collection in some late-stage pre- or post-marketing studies of drugs whose safety profile is well established, particularly with commonly occurring adverse events. By tailoring the method of safety data collection, it is anticipated that clinical trials can be conducted with greater efficiency, facilitating the conduct of large-scale trials designed to answer important questions on the efficacy and/or safety of a drug. The E19 Expert Working Group was established in 2017 with endorsement of the final concept paper by the ICH Management Committee. Over the course of the next 5 years, this expert work group met in Switzerland in 2017, Japan and the United States in 2018, ultimately issuing the draft guideline for public consultation in 2019. After receiving extensive comments seeking clarity on what is meant by selective safety data collection, raising concerns on how patient safety can be maintained, and requests for examples of how selective safety data collection can be employed, the expert work group met again in Singapore in November 2019 and continued to work virtually over the course of the next 2 years to address many of the themes raised during the public consultation period. and I am pleased to share with you the links to the finalized ICH E19 guideline published on September 27, 2022, and the FDA E19 final guidance issued on December 5, 2022. The guideline is not a lengthy document. It is comprised of 6 sections, and the main principles are captured in approximately 10 pages. The principles of E19 outlined in this guideline are intended for interventional clinical trials. More often, post-approval trials where experience from pre-approval trials can contribute to the knowledge of the drug's safety profile. However, in some circumstances, E19 may be considered for pre-approval trials. E19 is not applicable to gene therapy or to rare orphan disease clinical trials. And finally, implementing selective safety data collection as recommended in this guideline does not alter local regional safety reporting requirements. Ensuring the safety of trial participants is an important aspect of any clinical investigation. Safety monitoring in a clinical trial serves 2 important purposes. First, to protect the safety and well-being of individual trial participants. Second, to obtain safety information to be used in the assessment of the risk profile of the investigational medicinal product. There may come a time in the life cycle of a drug where a large amount of safety information has been evaluated and the risk profile of the drug is well characterized. Continuing to collect the same information will not add more to the knowledge of the risk profile of the drug, and selective safety data collection could be considered for further development of the drug. This doesn't necessarily contradict the first point made on the slide, because this guideline does not remove the responsibility of the investigator or the sponsor to ensuring the study participants' safety in the trial. When is a safety profile of a drug well established for us to consider selective safety data collection in a clinical trial? This guidance lists several factors contributing to this knowledge, but the list should not be considered exhaustive or determinative. First, what is the regulatory status of the drug? In particular, has the drug already been approved and is it marketed? What are the mechanistic factors? Is there a good understanding of how the drug works, its target of action, and is it the first in class or one of many where we have a lengthy experience with other drugs in that class? Is the clinical safety database of the drug an extensive one? Do we have large numbers of patient experience for an appropriate duration, and was the safety monitoring adequate in these programs? How similar is the planned trial where selective safety data collection is being considered to the previous trials? Do we have a very good understanding about the clinical pharmacology of the drug, especially drug-drug interactions, its metabolisms, and other factors that might also alter drug levels? Is there an extensive non-clinical program? And if the drug is already marketed, is there high quality and extensive post-marketing safety data? Once the safety profile of a drug is considered well established, what do we mean when we say a trial could implement selective safety data collection? In this guideline, selective safety data collection refers to the recording of certain data on case report forms by investigators, as well as reporting to sponsors for subsequent evaluation and submission to regulatory authorities. Generally, we would expect the following to be collected, even in a program with selective safety data collection implemented: serious adverse events, important medical events, medication error or overdose, whether they be intentional or unintentional, adverse events leading to study drug discontinuation, pregnancy and lactation exposure and outcomes of such exposures, and adverse events of special interest, including laboratory abnormalities identified in the protocol. Once agreement is reached between a sponsor and the regulatory authority that selective safety data collection is appropriate, collection of certain information may be limited or reduced in frequency of collection and this should be spelled out in the protocol. Some examples include non-serious adverse events, some lab monitoring, physical exams and vital sign collection, concomitant medications. All these could be limited in their collection. However, if an adverse event of special interest, important medical event, or an adverse event resulting in study drug or trial discontinuation occurs, a sponsor may need to collect the above information for that particular individual patient to characterize the particular event in more detail. It is important to note that for some of these with the asterisk, such information should be collected at baseline as part of comprehensive baseline evaluation before a patient receives study drug. Now let's discuss the possible situations where selective safety data collection may be considered. The guideline describes 4, although others might be considered, and in all cases, agreement would need to be reached between the sponsor and the regulatory agency. First, you might have an approved drug seeking new indication in a similar population to the one that is already approved. An example here could be an antithrombotic drug approved to reduce risk of heart attack and stroke in patients with existing coronary artery disease. Now, that same drug is being studied to reduce risk of heart attack and stroke in patients with peripheral artery disease. You could also have an approved drug seeking to expand its label to include additional endpoints in the same patient population. An example here could be a drug that was approved to treat heart failure based on a large outcomes trial showing reduction in risk of heart attack and hospitalization for heart failure. Now the company wants to study that same drug in the same type of patient population, but to evaluate heart failure symptom relief based on patient-reported outcome. You can have a safety trial with an objective to investigate specific safety concern. An example here would be a post-marketing required trial to evaluate a specific safety signal identified in the pre-market program. The benefit-risk profile of the drug was deemed favorable for approval, but the regulatory agency expects additional studies to further understand a specific safety signal, such as liver safety or cognitive impairment. Such a study is designed with a primary objective to evaluate that particular safety signal. You can have a trial designed to provide additional evidence of efficacy when current available data support a well-characterized safety profile. An example here would be a drug that was reviewed for marketing. No safety concern was raised that precluded approval, but one of 2 trials failed to meet statistical significance on a primary efficacy measure, and as a result, the drug was not approved, and the company is told to conduct another trial to establish clinical benefit. This guideline also describes possible methodologies for implementing selective safety data collection. You could have selective safety data collection in everyone. You could have comprehensive collection for a specific subset, for example, patients who are 65 years of age or older, and selective safety data collection in the rest. There could be comprehensive collection in everyone initially, but selective safety data collection thereafter. For example, a large cardiovascular trial was initiated in the pre-market period, and upon approval, the regulatory agency deemed that the safety profile was well characterized. This large cardiovascular outcomes trial would be ongoing at time of approval, but then could switch to selective safety data collection thereafter. There could be comprehensive collection in a representative subset, but selective safety data collection for remainder patients. For example, a randomly selected number of study sites could have selective safety data collection, or a randomly selected number of patients from each study could have selective safety data collection. Any method considered for selective safety data collection should be described in the statistical analysis plan, and agreement with the regulatory authority should be reached. To wrap things up, some key items need to be considered before implementing selective safety data collection. First, is the safety profile of the drug well characterized? 2. Do the study objectives and design of the trial support selective safety data collection? And 3. Has agreement been reached with a regulatory agency on the protocol implementing selective safety data collection? In conclusion, ICH E19 guideline will have a significant impact on the feasibility and efficiency of clinical trials designed to yield important new medical knowledge and advance public health. And while we have reached an important milestone in finalizing E19, there is still work ahead of us. Remember, drug development is a global endeavor. Therefore, alignment between regulators and industry is essential to advance principles laid out in this recently finalized guideline. There should be a concerted effort in the guidelines implementation, and we should share experiences and lessons learned as protocols and trials begin implementing E19. On behalf of the E19 Expert Work Group, pictured here from our meeting in Charlotte, North Carolina in November 2018, I would like to thank you for your attention, and I look forward to our question and answer session. Thank you.
Thank you for the great presentations. We'll now enter into our break time and resume in 10 minutes. Enjoy your break. Welcome back from the break. We hope you're able to get something refreshing. We're going to move right into our next presentations. Our first presentation in this session is on the updates on ICH safety-related guidelines, S1B-R1, rodent carcinogenicity studies for human pharmaceuticals, and M7R2, assessment and control of DNA reactive impurities in pharmaceuticals to limit potential carcinogenic risk by Dr. Elisa Vespa, Senior Scientific Evaluator and Pharmaceutical Drugs Director at Health Canada. And our second presentation is on S12, Biodistribution Studies for Gene Therapy Products by Dr. Sharon Choi, Senior Scientific Evaluator and the Biologic and Radiopharmaceutical Drugs Director at Health Canada. Please join me in welcoming our first presenter, Dr. Vespa.
Good afternoon. Today I will provide an update to two ICH guidelines. The first update will be for the ICH M7 guideline on the assessment and control of DNA reactive mutagenic impurities in pharmaceuticals to limit potential carcinogenic risk. And the second update will be on the ICH S1B guideline on testing for carcinogenicity of pharmaceuticals. Beginning with ICH M7, this guideline is currently composed of two parts, the main guideline and an addendum. The purpose of the main guideline is to provide harmonized guidance and a framework for the assessment and control of mutagenic impurities in pharmaceuticals to limit potential carcinogenic risk. The main guideline was adopted by ICH in June 2014. At present, the addendum contains monographs for 14 mutagenic and carcinogenic compounds commonly used in pharmaceutical manufacturing. As part of each monograph, a compound-specific acceptable limit is derived, and the availability of these limits facilitates consistency within the industry and across regulatory regions. The addendum was adopted by ICH as part of the ICH M7 guideline in May 2017, and this is the current Revision 1 or R1 version of the guideline. Currently, the ICH M7 Expert Working Group is in the process of finalizing the Revision 2 or R2 version of the guideline. For this update, several changes will be made and includes modifications to the main guideline, the addition of monographs and acceptable limits for 7 new compounds, as well as the inclusion of a new questions and answers or Q&A document to clarify recommendations in the main guideline that were unclear or led to different interpretation by stakeholders. Beginning with the main guideline, the most notable change is an update to Note 7. As background, the ICH M7 guideline indicates that the acceptable limit for a mutagenic impurity can be adjusted in an upward manner based on the treatment duration of the drug product. Note 7 of the ICH M7 guideline provides examples of clinical use scenarios for each treatment duration category defined in the guideline. In the current version of the guideline, HIV is listed as an example in the greater than 1 year to 10-year treatment duration category. However, because of advances in the clinical treatment of HIV, patient life expectancy is now close to normal. For this reason, HIV was moved into the greater than 10 years to a lifetime treatment duration category. Therefore, this change is associated with a reduction to the acceptable limit for a mutagenic impurity in a drug product that is used to treat HIV, going from 10 micrograms per day down to 1.5 micrograms per day. To avoid disruption to the supply of HIV drugs already on the market, a timeline for when this change comes into effect is provided in the ICH M7 question and answer document. Moving on to the ICH M7 addendum, this slide lists the mutagenic and carcinogenic impurities that will be added to the addendum, and they include 1,2-dibromoethane, epichlorohydrin, ethyl bromide, formaldehyde, styrene, acetaldehyde, and vinyl acetate. For each impurity, the available mutagenicity and carcinogenicity data is described in a monograph, and a summary of the methodology used to derive the compound-specific acceptable limit is provided. This slide summarizes the final acceptable limits for each of the 7 new impurities. There is insufficient time to go over the monographs in this presentation. However, once the addendum reaches step 4 of the ICH process and is published on the ICH website, it can be consulted for additional details. Of note, following the regulatory consultation, no changes to the proposed acceptable limit for any of the 7 new impurities, as described in the Step 1 document, were made. That is, the proposed limits will become the final limits. In regards to the monographs, changes were mainly editorial in nature to provide more clarity and in some cases context to the available data. Changes were also made to provide more consistency in terms of language used and with respect to formatting. Finally, Note 3 was added to the formaldehyde monograph, explaining how to calculate the acceptable concentration limit for formaldehyde in cases where it is present as an impurity in the API or drug product and the drug product is administered by the inhalation route and the concentration limit of 215 parts per billion in air is applied. At present, the ICH M7 guideline and the addendum are in the same document, as the addendum consists of Appendix 3 of the main guideline. However, due to the growing length of the addendum, now at approximately 150 pages, it will be removed from Appendix 3 and placed in a separate document. Appendix 3 will then contain a summary table which outlines the limits for each of the 21 compounds in the addendum, along with a high-level comment regarding methodology used to derive the acceptable limit. Importantly, these documents will remain connected, both on the ICH website and also with the use of hyperlinks within the documents themselves. The last part of this ICH M7 guideline update is the development of a questions and answers document by the expert working group. Since implementation of the ICH M7 guideline, it was noted that some of the safety and quality recommendations require clarification and inclusion of additional details to facilitate more consistent interpretation of the recommendations by stakeholders and to enhance harmonization across the ICH regions. Of note, stakeholders submitted more than 100 questions for consideration by the expert working group. Following an assessment of the questions and consolidation of related questions, a total of 25 questions and answers will be included in the final document. The Q&A document is structured in the same manner as the ICH M7 guideline and consists of sections 1 through 9. This slide outlines the title of each section of the Q&A and the number of questions developed for that particular section of the guideline, which includes any notes associated with that section of the guideline. Following the regulatory consultation, no changes to the proposed recommendations were made. Most changes were editorial to provide more clarity and sometimes context to the recommendations. Currently, the main guideline, addendum, and Q&A document are in Step 3 of the ICH process, which consists of regulatory consultation, expert working group discussion, and document revision. It is anticipated that these documents will be finalized and adopted as part of Step 4 of the ICH process in the near future, and once Step 4 is reached, the 3 documents will be posted on the ICH website. That concludes the update on the ICH M7 guideline. I will now move on to the ICH S1B guideline on testing for carcinogenicity of pharmaceuticals. The purpose of the ICH S1B guideline is to provide guidance on approaches for evaluating the carcinogenic potential of a pharmaceutical when carcinogenicity studies are warranted, per the recommendations in the ICH S1A guideline on need for carcinogenicity studies. The S1B guideline was adopted by ICH in July 1997. This slide will summarize at a high level the recommendations for how the carcinogenic potential of a pharmaceutical has been evaluated per the ICH S1B guideline, and it consists of one of two options. Under option 1, a 2-year carcinogenicity study is conducted in one rodent species, and a short-term or medium-term study is conducted in a second rodent species. What is generally observed when this option is used is that a 2-year study is conducted in the rat alongside a short-term study in a transgenic mouse model of carcinogenesis, such as the RAS-H2 model. Alternatively, carcinogenic potential can be evaluated using option 2, where a 2-year carcinogenicity study is conducted in 2 rodent species, most typically the rat and the mouse. Since the time the S1B guideline was published in 1997, a number of retrospective analyses were conducted, which suggested that in some cases, a 2-year rat study may not have been required to assess human carcinogenic risk of a pharmaceutical, and a comprehensive assessment of available biological, pharmacological, and toxicological data may have been sufficient. While the retrospective assessments were considered informative, it was unclear whether an evaluation of the available data would be sufficient to prospectively conclude that a 2-year rat study does not further inform human carcinogenic risk. For this reason, the expert working group proposed to conduct a prospective evaluation study to better define the factors that should be included in this comprehensive assessment, also known as a weight of evidence assessment, and also to determine the set of criteria that could support a conclusion that a 2-year rat study would not add value to the assessment of human carcinogenic risk with a reasonable degree of certainty. The concept paper and business plan describing the expert working group's proposal was developed and finalized in November 2012. And the prospective evaluation study was launched in the year 2013. There is insufficient time in this presentation to go over the design and results of this study, and so I refer you to the ICH website where the regulatory notice document, or R&D, is posted that provides details on the objective and design of the prospective evaluation study. Several status reports are also posted that provide a high-level summary of data obtained at specific points in time. Between the years 2013 and 2020, the study was carried out. Data was accrued and analyzed and discussed, leading to the development of the Step 1 draft addendum, and consensus was reached by the expert working group in March 2021. The draft addendum was endorsed by the ICH Assembly in April 2021. The document then proceeded through Step 3 of the ICH process, which consists of regulatory consultation, expert working group discussion, and document revision, and this step was concluded in July 2022. The addendum was then finalized and adopted as part of the ICH S1B guideline in August 2022. The updated guideline is now available on the ICH website at the web address outlined on this slide. The purpose of the S1B addendum is to expand the options for assessing human carcinogenic risk of pharmaceuticals by including a weight of evidence approach to determine if the 2-year rat study adds value to the assessment. It is important to note that the addendum does not replace the existing S1B guideline, and the Option 1 and Option 2 testing schemes discussed a couple of slides ago remain an important part of the carcinogenicity testing paradigm. In addition, the scope of the addendum does not include biotechnology-derived pharmaceuticals. For these compounds, the ICH S6 guideline should be consulted. The addendum also adds a plasma exposure ratio endpoint for high-dose selection in the RAS-H2 transgenic mouse model. This pharmacokinetic endpoint can be considered in addition to the other high-dose endpoints described in the ICH S1C guideline on dose selection for carcinogenicity studies. This is all I'm going to say about the plasma exposure ratio endpoint, as the main addition described in the addendum is the carcinogenicity weight of evidence approach. It is outlined in the addendum that the conduct of a weight of evidence assessment may lead to one of three possible conclusions. The first is that the carcinogenic potential of a pharmaceutical in humans is likely. In this case, the outcome of a 2-year rat study will not add value to the assessment of human carcinogenic risk. The second possible conclusion is that the carcinogenic potential of a pharmaceutical in humans is unlikely. In this case, the outcome of a 2-year rat study will also not add value to the assessment of human carcinogenic risk. The third possible conclusion is that the carcinogenic potential of a pharmaceutical in humans is uncertain. In this case, the outcome of a 2-year rat study will add value to the assessment of human carcinogenic risk. So what factors should be evaluated in the weight of evidence assessment to determine whether or not the conduct of a 2-year rat study will add value to the assessment of human carcinogenic risk? In total, 6 primary weight of evidence factors are outlined in the ICH S1B addendum. The first is an understanding of potential carcinogenic risk based on drug target biology and the primary pharmacologic mechanism of the parent compound, as well as any major human metabolites. Assessment of this factor will likely consider information from various sources, and examples are outlined in the addendum, which includes carcinogenicity data from other compounds in the drug class. The second factor is secondary pharmacology to evaluate off-target potential, which can include the results of a secondary pharmacology screen for the parent compound and any major human metabolites. Of note, assessment of off-target effects relevant to potential carcinogenic risk may be considered particularly informative and can include, for example, a nuclear receptor screen. The third factor consists of an evaluation of histopathology data from repeat dose toxicity studies. Histopathology data from the long-term rat toxicity study is the most informative for assessing the value of the 2-year rat study. The fourth factor consists of evidence of hormonal perturbation. Sources of data for this factor are various and can include histopathology findings in endocrine and reproductive tissues, as well as the results of reproductive toxicology studies. The fifth factor consists of genetic toxicology data derived from studies outlined in the ICH S2 guideline on genotoxicity testing. And the final factor consists of evidence of immune modulation, which can include results from studies outlined in the ICH S8 guideline on immunotoxicity. In some cases, comprehensive assessment of these 6 weight of evidence factors may be sufficient to conclude that a 2-year rat study is or is not likely to add value to the assessment of human carcinogenic risk. However, in other cases, data from investigative studies may be needed to better inform the human relevance of an identified risk, and prior to concluding whether or not a 2-year rat study will add value to the assessment. The ICH S1B addendum indicates that both non-clinical and clinical approaches can be used. In regards to non-clinical approaches, the following are examples provided in the S1B addendum and include special histochemical stains, molecular biomarkers, measurement of serum hormone levels, assessment of immune cell function, and data from emerging technologies. In regards to clinical approaches that can be used, the goal is to determine human mechanistic relevance at therapeutic doses and therapeutic exposures, and can include, for example, measurement of clinical plasma hormone levels. Once all the relevant data for each weight of evidence factor and any additional investigative studies is gathered and assessed, the information is integrated to determine whether or not a 2-year rat carcinogenicity study will add value to the assessment of human carcinogenic risk. How this integrated assessment may be accomplished is outlined in the form of case studies drawn from the prospective evaluation study and are presented in Appendix 1 of the addendum. Section 2.3 of the addendum provides recommendations in regards to mouse carcinogenicity studies. Importantly, a mouse carcinogenicity study remains a recommended component of the carcinogenicity testing plan, even if the outcome of the weight of evidence assessment indicates that a 2-year rat study does not add value. The mouse carcinogenicity study can be conducted as a 2-year study in a standard strain of mice or a short-term study in a transgenic model. However, the addendum does indicate that a mouse carcinogenicity study may not be appropriate in some cases, such as when the weight of evidence assessment indicates that the compound is likely to be carcinogenic in humans. The outcome of the Step 3 regulatory consultation on the Step 1 draft addendum did not lead to any substantial changes to the recommendations. Several editorial changes were made to improve flow and to provide clarity in regards to the recommendations. In addition, 2 figures were added to the addendum to visually represent how the weight of evidence approach is carried out. This is the first figure that was added, and its purpose is to summarize the key steps leading to the development of a carcinogenicity assessment strategy. So the first step is to gather data for each weight of evidence factor. Once the data has been gathered and analyzed, it is integrated so that a conclusion can be reached regarding the likelihood that a compound exhibits carcinogenic potential in humans. As previously mentioned, there are 3 possible conclusions. Carcinogenic potential in humans is likely, unlikely, or uncertain. The carcinogenicity assessment strategy associated with each of these conclusions is outlined at the bottom of the flow scheme. This flow scheme also visually demonstrates that the selected carcinogenicity assessment strategy should be based on a thorough assessment of relevant data, even if the outcome leads to the conclusion that a 2-year rat study will add value to the assessment of human carcinogenic risk. This second figure was added to facilitate how the data across the 6 weight of evidence factors is integrated to determine if a 2-year rat study will or will not add value to the assessment of human carcinogenic risk. At a high level, if the characteristics of a compound align more towards the right-hand side of the figure, it is less likely that additional investigative studies and/or a 2-year rat study will be warranted. Whereas if the characteristics of a compound align more towards the left-hand side of the figure, it is more likely that the additional investigative studies and/or a 2-year rat study will be warranted. As an example, in regards to secondary pharmacology, additional investigative studies and/or a 2-year rat study will be less likely in cases where the compound exhibits high target selectivity and no off-target activity, and will be more likely in cases where the compound exhibits low target selectivity and off-target activity. The figure also demonstrates that, in some cases, additional investigative studies may be conducted to further inform any identified concerns from the 6 primary weight of evidence factors, and both non-clinical and clinical approaches can be used. The last outcome of the regulatory consultation to highlight is that a new recommendation was included in Section 2.3 on mouse carcinogenicity studies, and it states that use of a transgenic model is consistent with the 3R principles, and this model should be prioritized unless there is a scientific rationale for conducting a 2-year study in mice. And that concludes the update on the ICH S1B addendum. As mentioned, the updated S1B guideline, which contains the addendum, is now posted on the ICH website and can be referred to by all stakeholders. This is my final slide, and I would like to acknowledge members of the ICH M7 and ICH S1 expert working groups, as all of the information presented here are work products of these groups. And with that, I would like to thank you for your attention.
Hello everyone, and thank you for joining me today. My name is Sharon Choi, and I am a Senior Scientific Evaluator at Health Canada, in the Biologic and Radiopharmaceutical Drugs Directorate. Today I'll be presenting on the new ICH guideline, ICH S12, on non-clinical biodistribution considerations for gene therapy products. This is the first ICH safety guideline on gene therapy products. As we all know, gene therapy is a rapidly growing field, and so there's been a need for harmonizing aspects of the non-clinical development program for these products, especially given that various jurisdictions have their own guidance documents on the topic, including their own definitions of what a gene therapy product is and what constitutes biodistribution. The need for this gene therapy guideline in particular is based on the importance of first characterizing the biodistribution profile of a gene therapy product to inform the design of subsequent non-clinical pharmacology and especially toxicology studies, which in turn are conducted to support early phase clinical trials. In terms of the timeline for this guideline, we're currently in Step 3 at the Expert Working Group sign-off stage, with the aim of reaching Step 4 guideline adoption very shortly. The purpose of this presentation today is to provide an overview of the main points in the guideline, to start the main sections as laid out in the table of contents here, and in the guideline are 1, the introduction to the guideline, 2, definition of non-clinical biodistribution or BD, 3, timing of non-clinical BD assessments or studies, 4, design of non-clinical BD studies, 5, specific considerations for BD assessments, and 6, application of non-clinical BD studies. To start, the overall objective of the ICH S12 guideline is to provide harmonized recommendations for non-clinical BD studies and assessments for gene therapy products. And so the guideline provides specific recommendations for the overall design of non-clinical BD studies while keeping in line with the 3 R's principles of reducing, refining, and replacing the use of animals. The guideline also provides specific considerations for the interpretation and application of the BD data that are generated from these studies. We'll first start off with how gene therapy products and the term biodistribution are defined within the guideline. For the purposes of this guideline, gene therapy products are defined as therapeutic products that mediate their effect by the expression of transferred genetic materials or by specifically altering the target genome of human cells. Now, examples of gene therapy products that fall within this definition include purified nucleic acids, microorganisms that are genetically modified to express transgenes, and ex vivo genetically modified human cells. Now, products that are excluded from the scope of this definition and therefore the scope of this guideline include prophylactic vaccines and chemically synthesized oligonucleotides. Also, for the purposes of this guideline, non-clinical biodistribution is defined as the in vivo distribution, persistence, and clearance of a gene therapy product at the site of administration and in target and non-target tissues, including biofluids. So it's a fairly classical definition of what distribution is. Now, excluded from the scope of this definition and from this guideline are the topics of shedding and genomic or germline integration. Discussions on these topics, though, can be found in existing ICH consideration documents. In terms of the timing of biodistribution assessments, so when these should be conducted, the data from BD assessments should be available to allow for the interpretation of pharmacology and toxicology findings, and should also be completed prior to the first in-human clinical trial. In the next couple of slides, I'll go over the study design recommendations for BD studies that are laid out in the guideline. So first, such studies can be conducted as standalone studies, or they can be combined with a pharmacology or toxicology study. Also, BD studies can be conducted as GLP compliant or non-GLP compliant studies. Importantly, though, if they're conducted as a non-GLP compliant study, the study is still expected to be of good quality in order to ensure data integrity and reliability. Another thing to note is that if the BD assessment is done as part of a GLP compliant study, such as a GLP compliant toxicology study, the sample analysis can be done in a non-GLP compliant manner, but it remains important that the in-life evaluation and sample collection procedure are still done in compliance with GLP. With respect to administration of the product, this should reflect the intended clinical use. So the test article used should be representative of should be representative of the intended clinical product, and the route of administration should be the same as the clinical route of administration. The doses selected to be administered in the animal should be equal to or greater than the anticipated maximum clinical dose, with the highest dose preferably being the expected high dose used in the toxicology studies. So essentially the doses that are selected for administration in the animal studies should be relevant and enable interpretation of the pharmacology and toxicology studies. The study design recommendations also include that the species selected should be biologically relevant, or that the animal model selected should support transfer and expression of the genetic material. In terms of sex, both males and females should be included on study unless otherwise justified. For example, if the product is intended for use in females only, then including only female animals would be acceptable. For animal numbers, the animal numbers and group size should consist of an appropriate number of animals per sex per group per time point to generate sufficient and meaningful data. Note 2 in the guideline does provide more specific details on the number of animals, including the recommendation that a minimum of 5 rodents or 3 non-rodents per sex per group per time point be evaluated. And finally, in terms of tissue sample collection, tissue samples should be collected at multiple and appropriate time points that together can cover time-related changes in the gene therapy product levels in the tissues. So I've listed here tissues that should be collected, and these include blood, injection sites, gonads, adrenal gland, brain, spinal cord, liver, kidney, lung, heart, spleen, and then any other relevant tissues. This list can be expanded to include additional tissues based on background knowledge of the product and the intended clinical use, including the target disease population. So in addition, the sample collection procedure should be designed to minimize the potential for contamination. So it's important to follow a pre-specified process. For example, one would collect control samples first and then test article samples in ascending order of dose. Now, there are also other specific or special factors that should be considered when designing a BD study or when performing a BD assessment. These specific considerations are listed out in the following 2 slides and in section 5 of the guideline. First, the assay should be one that quantitates the amount of the genetic material itself. Quantitative PCR is currently the gold standard, but other assays can be used, and the guideline does provide room for the use of evolving technologies. For measurement of expression products, now expressed products can also be measured and may be encouraged, as these data can contribute meaningful information for the characterization of the safety and activity profiles of the product. Immunological concerns should also be considered and addressed. For pre-existing immunity concerns, animals should be screened, and if possible, only negative animals should be included on study. Immune responses to the gene therapy product or to the expression product can also occur after administration in animals, which would result in data that might not be informative due to increased clearance of the product or the expression product. So in the case of an immune response to the gene therapy product, immunogenicity analysis should be performed to aid in the interpretation of the BD data. In the case of an immune response to the expression product, one might consider using a species-specific transgene instead to avoid the consequences of the immune response in the animal. For ex vivo genetically modified cells, factors such as cell type, route of administration, and the potential for the expression product or gene modification event to affect the expected distribution of the cells should be considered. As mentioned previously in the study design slides, gonadal tissue should be collected for analysis and should include both male and female gonads. It should also be considered whether there is persistent presence within the gonads, since persistence can trigger the need for additional studies to determine which cell types in the gonads are transduced. You can refer to the ICH considerations paper on this topic for further guidance. On the topic of what would trigger the need for additional non-clinical BD studies for products that have existing BD data and have gone through clinical trials and or authorizations. In this case, additional BD studies may or may not be needed. Additional studies would be needed if there are significant changes to the product or conditions of use, such as changes in the route of administration, an increase in dose, more frequent dosing regimen, changes in the vector structure or serotype, or other significant changes in the manufacturing process, which would include changes in the final formulation and changes in the properties of the final product. Otherwise, additional studies would likely not be needed. Existing BD data can support the additional indications for the same product, but again, all the factors that I just mentioned would still need to be considered first. Last is the subject of application of non-clinical BD studies. So how do we apply the data generated from BD studies? In other words, why are these data important? As I mentioned at the beginning, non-clinical BD data are important in the development of a gene therapy product, and this is because they contribute to the interpretation and design of subsequent non-clinical pharmacology and toxicology studies. And they can also inform elements of clinical trials, including safety monitoring plans. So this brings me to the summary of my presentation. So in summary, ICH S12 is the first non-clinical ICH guideline on gene therapy products and provides for a harmonized definition for gene therapy products, recommendations on the timing and optimal design of BD studies, factors and potential effects to consider when designing BD studies, and factors to consider when determining the need for BD studies. And that brings me to the end of the overview of the ICH S12 guideline. I'd like to take this time to acknowledge all of the S12 expert working group members, including Dino Pichin from Health Canada, our colleagues at FDA, and our rapporteur Masakazu Harada from the PMDA. Thank you very much for your attention.
Thank you all for the great presentations. We're on the home stretch now. Our final two presentations for the day provide updates on ICH quality-related guidelines. First presentation is on Q5A-R2, Viral Safety Evaluation of Biotechnology Products Derived from Cell Lines of Human or Animal Origin by Chris Storbeck, who's with the Biologic and Radiopharmaceutical Drugs Director at Health Canada. And our final presentation is on Q13, Continuous Manufacturing by Dr. Larry Lee, Deputy Director of Science in the Office of Pharmaceutical Quality, CDER FDA. That'll leave us right into our Q&A panel, which will be moderated by Nick Orphanos and Jill Adelberg. Jill serves as FDA's ICH coordinator, acting as a liaison between ICH and over 100 FDA scientific experts working on dozens of harmonized and international drug regulation standards. She's worked at FDA for over a decade. Prior to joining CDER as ICH coordinator, Jill advised senior agency officials on major health legislation, including the 21st Century Cures Act and reauthorization of the Prescription Drug and Animal Drug User Fee Acts. During prior stints at CDER and CDRH, Jill helped to launch the Generic Drug User Fee Program and the Medical Countermeasures Initiative. Now, please join me to welcome our first presenter in the final session, Chris Storbeck.
Good afternoon. It's a pleasure to be here to talk about the ICH Q5A revision for viral safety evaluation of biotechnology products derived from cell lines of human or animal origin. I'll begin by providing an outline. I'll give some background into the history of the revision. I will then follow up with some key principles that were discussed and maintained in the document. I will then go into key updates that were made to the document to reflect some of the important changes that were introduced. And finally, I'll end with a summary. So ICH Q5A-R1, the original document, was finalized in 1999. In 2019, the concept paper and business plan discussing the proposed changes was endorsed in Singapore. And the Step 2 document, the revised draft document, was signed off last fall. And this document was issued by the ICH regulatory members for public consultation at that time. And currently, the expert working group is going through the comments that have thus far been received, and we're anticipating finalization and implementation of the Step 4 document in the fall. So the consensus among the expert working group from the beginning of the process was that the original document remains very useful. However, given the advances in biotechnology and scientific advances of the past 20 years, it was determined that there's a revision is necessary to reflect updates in manufacturing processes that are not yet captured with respect to virus risk, like continuous manufacturing. New product types that have been evolving in the last 20 years are now going to be captured by the guidance, such as gene therapies. Also, new analytical technologies that may improve the measurement of virus contaminants, such as next generation sequencing, is introduced. And alternative virus clearance validation strategies that are allowed to come into being based on the evolving scientific knowledge of the last. So there's been a recognition that the original structure and the key principles of the document function very well, and there's that has been agreed to be preserved as the document goes forward. The focus will be maintained on requirements for marketing authorization, and the key scientific principles will be highlighted. However, the flexibility of the original document will be retained, and this is to continue to allow for scientific evolution. And where scientific consensus exists, this will be described. So the key updates introduced focus on 6 key areas. So I mentioned the new product types and the manufacturing changes, continuous manufacturing, and also new test methodologies. Also, advancing scientific knowledge has indicated that there can be new approaches with regard to resin reuse for certain chromatography resins. Prior knowledge has made allowance for the application of platform virus clearance data in some situations, and also for certain well-characterized rodent cell lines, there can be some flexible approaches in terms of some aspects of their use. I'll get into those a bit later. And finally, with the introduction of all of these changes to the document, the glossary requires updating with some new terminology. So I'll begin with the new product types. So the scope has been broadened to encompass genetically engineered virus vectors. So these are products where a virus is the product, and they have to be amenable to virus clearance. This is now detailed in the scope. And in some cases, for example, with AAV, helper viruses are used in their production. Also, products that are manufactured using viruses, but the virus isn't the product itself. The virus is just used to generate the products, such as the manufacture of virus-like proteins using a baculovirus insect cell system. And new aspects of the guidance deal with the baculovirus as a contaminant and the insect cells as a new cell substrate that can present certain risks. Also, nanoparticle-based vaccines and therapeutic products are incorporated into the scope of the guidance. And a new Annex 7, has been introduced with respect to new product types, providing some discussion of those new product types and the testing requirements. So sections that have been updated to reflect the new product types include Section 2, where there's reference to the new product types, Section 3, where there's recommended virus detection and identification assays. In Section 5, a new case has been added, Case F, and Table 4 has been updated to reflect the dealing of helper virus used in production. And as mentioned, Annex 7 has been added to the document, and this is to detail genetically engineered viral vectors and viral vector-derived products. So continuous manufacturing has been added, and it's for the viral clearance considerations that come with continuous manufacturing. And a new section, Section 7, has been added with regard to this. Continuous manufacturing can mean a fully continuous process or a subset of unit operations run in a continuous fashion, and the remainder is operated similar to a batch process. So the guidance focuses on unique viral safety concerns that are presented with continuous manufacturing, not present with batch mode. And also, the new elements for continuous manufacturing should be considered in conjunction with ICH Q13, which is a guidance document dedicated to continuous manufacturing. So modeling continuous manufacturing can be challenging, and leveraging experience with batch mode at a small scale for certain virus clearance unit operations can be helpful. The guidance focuses on risks that are specific to continuous manufacturing, such as the length of time in culture. For example, there can be fluctuations in the levels of endogenous retroviruses, the element of diversion or segregation if material in the event of potential contamination or other failure of the process. This must be considered. In addition, risks associated with the integration of unit operations, such as using tandem chromatography steps, must be addressed. Furthermore, sampling considerations, there must be appropriate process monitoring and sampling strategies to detect inadvertent disturbances or adventitious agent contaminations is essential. Process output may vary during startup, termination, or during process disturbances. These possibilities may be addressed using the existing best practices for virus clearance studies. Considerations specific to CM include chromatography. Virus clearance validation as a batch process could serve as a scaled-down model when a multi-column is used as repeating sub-batches. Additionally, validation of virus clearance could be performed as a single unit operation or with two columns operated in tandem. For low pH, virus inactivation validation as a batch process could be appropriate with well-justified target process conditions that ensure critical process parameters are maintained. For vial filtration, virus clearance as a batch process may also be appropriate so long as the process parameters remain within worst-case set points. Also, process controls must allow for filter changes, post-use integrity testing, and allow for diversion. Next generation, or high-throughput sequencing, is introduced in the revision because of its broad detection, high sensitivity, and flexibility in terms of application of the technology. It can be used agnostically, or it can be used in a targeted fashion. It's been encouraged to replace the in vivo test. However, justification should be provided and also a validation package to ensure that it's fit for purpose. It may also be used to supplement or replace the in vitro test. The molecular methods are also encouraged to replace the HAP, MAP, and RAP methods. So using methods such as targeted NGS or multiplex PCR. Now, the replacement of the in vivo methods with molecular methods is in line with the 3 Rs initiative of the EMA or EU, and despite the encouragement to use the molecular methods, the flexibility of the document remains, and the in vivo method may still be applied. With the evolution of scientific knowledge in the last 20 years regarding affinity capture resins, with the realization that there's no decline in virus clearance capability of the protein A resins with repeated use. There's a line specifically indicating in the guideline that product-specific end-of-life studies are not required. This may be applicable to other column types. However, specific product knowledge, equivalent prior knowledge, including in-house experience, and a detailed justification would be required in place of specific viral clearance studies. The use of platform virus clearance and activation in place of product-specific virus clearance validation may be acceptable. The platform process must be well characterized. There must be an understanding of the mechanism of virus clearance and inactivation. there must be the composition of the process intermediate must be highly similar, as this can impact the virus clearance capability. Additionally, there must be equivalence of the upstream step, as this can influence the presence of impurities that may also impact the virus clearance capabilities. And the robustness of the critical parameters must be demonstrated so as to ensure that the operation within those parameters ensures that the virus clearance is maintained. In support of this, a new Annex 6 is added to provide examples of permitted steps, such as the detergent solvent inactivation, low pH inactivation, or nanofiltration. In these examples, process parameters critical to the inactivation or removal of the virus are considered. In addition, flexible approaches for well-characterized cell lines has been introduced into the document. For example, a master cell bank in vivo test may be waived if there's a risk-based justification provided that details the cell type. For example, if it's CHO, NS0, or SP20. The testing is performed on the parental cell line and on multiple master cell banks derived from that same cell line. However, if residual risk remains, then further testing may be required. In terms of the safety factor for the log reduction value of CHO, in the initial guideline, the example given was a safety factor of 10 to the minus 6 particles per dose. This has been modified to 10 to the minus 4 particles per dose based on the non-infective nature of the retrovirus-like particles in the cell line, and also this must be accompanied by a negative in vitro test. So for CHO-derived products, and CHO retrovirus-like particles may be utilized as specific model viruses for virus clearance studies in this situation. In terms of the new additions to the revision, the glossary had to be refined or modified to account for and reflect the expectations for new product types, including helper virus, viral vector-derived product, master virus seed, and working virus seed. It's important to provide clear definitions for these different new being introduced. In addition, to reflect expectations for prior knowledge, new definitions have been provided for platform validation of virus clearance, process robustness of virus clearance, and prior knowledge. New definitions to align terminology include endoproduction cells. So in summary, the original guideline, the overall layout is preserved, as the functionality of that layout has been appreciated for 20 years since the inception of the original document, and also the flexibility has been preserved as well in order to accommodate future scientific and technological advances. So these updates to the ICH Q5A reflect advances in science, medicinal products, manufacturing processes, and testing technologies. I want to thank you for your attention. I'll be present for the panel discussion later on this afternoon, where I'd be happy to take any questions.
Good morning. I'm Larry Lee from FDA and also the rapporteur of ICH Q13. Today, I'm going to provide update regarding our progress on ICH Q13 on continuous manufacturing, CM, of drug substances and drug products. I'm happy to announce that the ICH formally adopted this CM-ICH guideline document in November 2022. This document was developed based on a concept paper and business plan approved in November 2018. Despite COVID, we Q13 expert working group were able to finish it in 4 years. In the context of Q13, we define continuous manufacturing CM as a process involving the continuous feeding of input materials into the transformation of in-process materials within and the simultaneous removal of output materials from a manufacturing process. Specifically, Q13 focuses on the integrated aspect of a CM system in which two or more unit operations are directly connected. Building on existing ISH quality guidelines, this ISH document describes fundamental concepts, scientific principles or approaches, and regulatory considerations specific to the development, implementation, operation, and life cycle management of CM for drug substances and drug products. The Q13 is intended to capture key scientific and regulatory considerations that promote harmonization, including certain CGMP elements specific to continuous manufacturing, as well as to provide guidance to industry and regulatory agencies regarding regulatory expectations on the development, implementation, and assessment of continuous manufacturing. To cover CM topic that is broad in scope, we organize the Q13 document into two parts. The first part focuses on fundamental aspects of continuous manufacturing that are generally not specific to technology, dosage form, or molecule type within the main guideline. In the second part, we use analysis to augment the main guideline by providing illustrative examples and considerations specific to certain modalities, technologies, and production methods. For main guideline, we have 6 sessions including introduction, continuous manufacturing concepts, scientific approaches, regulatory considerations, glossary, and references. We also have 5 analyses to cover specific considerations for continuous manufacturing of drug substances of chemical entities and therapeutic proteins, continuous manufacturing of finished dosage forms, and end-to-end integration of drug substance and drug products, as well as management of disturbances encountered during continuous manufacturing operation. The introduction defines the objective and scope of Q13. I have already described the objective of Q13 in the beginning. Here, I would like to point out again that the scope of Q13 is broad in nature and is also forward-looking, as it covers continuous manufacturing of drug substances and drug products for chemical entities and therapeutic proteins, even though the maturity of these continuous manufacturing technologies varies. Q13 is also applicable to development and implementation of continuous manufacturing for new drugs, including therapeutic proteins, generic drugs, and biosimilars, as well as the conversion of batch manufacturing to continuous manufacturing for existing products. Furthermore, the principles described in this guideline may apply to other biological and biotechnology entities. For continuous manufacturing concepts in Section 2, we list and explain different modes of CM integrations, including the partial integration of unit operations, where some unit operations are operating in a batch mode. full integration of a drug substance and drug product manufacturing process, and end-to-end integration with no isolated drug substance. We also clarify how to define the batch size based on the existing batch definition described in ICH Q7. For continuous manufacturing, the batch size can be defined in terms of quantity of output materials, quantity of input material, runtime at a defined mass flow rate, or other approaches if scientifically justified. For scientific approaches in Session 3, we focus on a holistic approach for control strategy, with emphasis on key continuous manufacturing control strategy elements, such as process dynamics, equipment design and system integration, process monitoring and control, material traceability and diversion, and process models. We also describe the risks associated with different approaches to change production output. We highlight that the increased use of real-time monitoring and control of process and quality attributes in continuous manufacturing, among other things, makes continuous process verification well suited to the evaluation of continuous manufacturing processes from a process validation perspective. In Session 4, we clarify the key aspects of regulatory expectations specific to continuous manufacturing. With respect to marketing application, post-approval changes, site implementation, and pharmaceutical quality systems. These key aspects are listed in these slides here. We also clarify what and where continuous manufacturing specific information should be included in the dossier according to the CTD. Our intent here is really to provide clear and harmonized guideline or guidance to industry on what continuous manufacturing information should be provided in a drug application in order to facilitate a more streamlined regulatory assessment of these new continuous manufacturing technologies. Meanwhile, Q13 will still allow industry to employ or use a flexible approach for continuous manufacturing development and implementation. The annexes serve many purposes in Q13. Annexes 1 and 2 provide examples of approaches to implement CM of drug substances for chemical entities and CM of immediate release solid oral drug products, respectively. The intent here is to use these examples to clarify or help illustrate the scientific principles discussed or described in the main guideline. In comparison, Annex 3 augments the new guideline, the main guideline by providing additional considerations specific to therapeutic protein drug substances and drug substances used as intermediates for subsequent conjugation. Annex 4 describes additional considerations unique to development and implementation of an integrated drug substance and drug product continuous manufacturing process for a small molecule tablet dosage form. Specifically, we focus on the technical and regulatory considerations related to the drug substance and drug products interface, where no drug substance is isolated prior to the finished dosage form manufacturing. NS5 provide examples of approaches that can be used to manage transient disturbances that may occur during operation. Our intent here is to point out that some disturbances may not have impact on product quality, and not all disturbances will lead to material disposal and investigation. The slide here shows the next steps and milestones. In January 2023, ICH adopted the Q13 Implementation Working Group concept paper for developing training materials, and the Implementation Working Group was established. The goal of this Implementation Working Group, IWG, is to prepare and deliver a training program that will help facilitate an aligned interpretation and harmonized implementation of ICH Q13 in ICH and non-ICH regions. The intent of this working group is not to provide comprehensive training on all aspects of continuous manufacturing, but rather to illustrate the application of specific concepts or principles of Q13. With this, I would like to conclude my presentation and thank you for your attention.
As Ray mentioned earlier, I'm Jill Edelberg, FDA's ICH coordinator, and I want to thank all of our experts for their excellent presentations. I'd also like to thank the audience for the great questions that we've received so far. Before we begin the questions and answer session, I want to let everyone know that Dr. Choi is unable to join us. In her place, we're delighted to welcome Dr. Alyssa Galaro. Alyssa serves as the FDA's topic lead on the S12 Expert Working Group and as the Pharmacology Toxicology Team Lead in the Office of Tissues and Advanced Therapies. We're also delighted to be joined today by Dr. Ron Fitzmartin and Dr. Lei Zhang. Ron recently took over the reins as Rapporteur or Scientific Lead of the M11 Expert Working Group, and he and Vivian are available to answer M11 questions. Ron serves as Senior Informatics Advisor at FDA's Center for Biologics Evaluation and Research. Lei serves as the Rapporteur for the M13 Expert Working Group and is a Deputy Director in FDA's Office of Generic Drugs, and is available with John to answer any M13 questions. My colleague Nick and I are going to moderate the Q&A session, and I'd like to begin by posing the first question to Nick. Nick, are ICH guidelines considered mandatory to follow?
Thanks very much for the question, Jill. This is actually a very good question and one we actually get quite often. So the short answer is that ICH guidelines are considered guidance documents once they are implemented by regulatory agencies, which are actually not considered legally binding unless they are cited in regulation. So with that said, they are not considered mandatory. However, all ICH members are extremely committed to the implementation of ICH guidelines, and we do strongly encourage stakeholders to follow them. They have been given in-depth consideration and discussion amongst ICH members, which include regulators and global industry associations. They obviously, as we've presented today, they align technical requirements globally and will therefore reduce regulatory stakeholders, especially those submitting products for approval in multiple regions. So that's it for that question. I will move on to pose the next question, which is for Dr. Anna Edmondson on ICH M10. So the first question for Dr. Edmondson is, should bioanalytical method validation be conducted under good clinical practice or good laboratory practice?
Thank you. That's a great question. The analysis of samples from non-clinical studies should be conducted according to the principles of GLP, or good laboratory practice, and the analysis of samples from clinical studies should be conducted according to the principles of good clinical practice, or GCP.
Thank you very much. Moving on to our next question. This one is for Vivian or Ron on M11. Once ICH M11 goes through step 4, will the protocol be required or recommended?
Thanks, Jill. It's a good question. So as Vivian mentioned, we are currently in step 3. When we move to step 4, our documents would be will have been adopted by the ICH regulatory members. But then there's a step 5. Step 5 is implementation by the regulatory members according to their local, regional, or national requirements. However, as far as FDA, as many of you know, under Section 745A of the FD&C Act, has the authority to require certain submissions to be submitted in a specified format. FDA has not, at this time, made a determination whether or not the ICH M11 documents fall under 745A. Thank you.
Thanks very much, Ron. And I turn it over to Nick.
Thanks, Ron and Jill, for that. The next question is for Raj on ICH M12. The question is, the draft guidance provides a list of drugs for in vitro and clinical DDI studies in the appendix. Is it possible to use drugs not listed in the appendix to perform some of the DDI evaluations?
Thank you, Ron. That's a very nice question. So when we put up these lists of drugs for both in vitro studies as well as for clinical DDI studies, these were not meant to be comprehensive and all-encompassing. Our effort was to make sure that we had at least one drug per category, which could be utilized across all the geographic regions. Having said that, we do state in the guidance that when a selective drug is available and it is selective at the concentrations for in vitro studies, that information can definitely be presented to justify the use of a drug which is not in that list. Similarly, for the clinical DDI studies, sponsors could select the drugs based on sensitivity, specificity, and safety profiles for those particular pathways. So in a nutshell, it's yes, but they should be justified when they are not listed as part of these drug lists in the appendix. Thank you.
Our next question is on M13 for John or Lei. The question is, although this was not mentioned in today's presentation, I note in the posted M13A guideline that in some situations it will be acceptable to employ a low-fat, low-calorie meal in a bioequivalence study that needs to be conducted under fed conditions. What is the rationale for this change from the usual high-fat, high-calorie meal for such studies?
Thanks, Jill. That is a very good question because this is a new element that appears in M13. I think to understand it, you first need to look at the value of the classic high-fat, high-calorie meal, which is typically used in fed studies. I think that it needs to be appreciated. The greatest value of that is that a high-fat, high-calorie meal, especially a warm meal, is thought to be providing the greatest deviation in GI tract conditions compared to what you would see under fasted conditions. So when you've got a study conducted under fasted conditions and a study that's conducted under high-fat, high-calorie conditions, you're sort of covering the whole spectrum of GI tract conditions that you might experience, that a patient might experience, depending on what they've eaten. So that's the value of that study. But in cases where there will only be one study conducted, so with respect to M13A, low-risk products, under certain conditions where the comparator is recommended to be taken with food. In this case, you are only going to have one study conducted. And so then the issue becomes, is is the scenario where you've got the greatest perturbation from fasting conditions really what you're looking for? Or is it more appropriate to look at fed conditions which are more moderate or more middle of the road, if you like, or more typical perhaps in many situations for patients? And so in those cases for low risk products, M13 guideline says that a low fat, low calorie meal could be employed, or a high fat, high calorie meal, depending on the choice. It should be pointed out, of course, though, if the comparator product labeling specifies a meal type, the product should be taken with this sort of meal, then that would be the meal that would be employed in that situation. Thank you.
Great, thank you, John, for that response. The next question is for Mary Tan Hai on ICH E19. The question is, can you elaborate more on whether selective safety data collection can be considered for rare diseases if the product is already approved for a highly prevalent condition?
Hi, great. Thanks for the question. That's actually an excellent question. You may recall that I've mentioned the scope of E19 didn't include for rare orphan diseases. And the reason why is that really to make the determination that the safety profile of a drug is well established, we certainly would want to make sure that we have had adequate clinical experience, actually not just clinical, but a large program that actually can provide the evidence that it's a well-established safety profile. And for rare diseases, the limited number of patients that can be exposed is likely not going to be able to contribute to that. But the question here is posing if the drugs are approved in a highly prevalent condition. And in that setting there, it's quite possible that the safety profile of the drug in prior indications can actually provide reasonable assurance that the safety profile is well established. And there could be a justification made to actually use selective safety data collection if now that drug is going to be studied in a rare disease condition. Some caveats there, though, that you need to make sure that the dosing regimen is the same, that actually the benefit risk profile, the risk in the population and the rare disease population, if it's a vulnerable population, there may be still some concerns that you couldn't entirely use selective safety data collection. I think the key point to make here is that if a sponsor felt that there is justification to use an already approved drug to study in a rare disease and that the safety profile of that already approved drug is well established, to submit that as a proposal to a regulatory authority and to get that agreement, discussion and agreement with the regulatory authority before initiating the trial. Thank you very much.
Thanks very much, Mary. The next question is for Elisa, and the question is, has any consideration been given to the volatility of formaldehyde in setting the acceptable limit?
Thank you for that question. Thank you for that question. So due to the volatility of formaldehyde, there was discussion within the expert working group regarding whether formaldehyde should even be included in the ICH-M7 addendum. And in the end, it was decided that a monograph and a limit would be developed for formaldehyde. And while the limit is based on the available toxicity data, there may be chemistry arguments to support a different control strategy. Thank you.
Great, thank you, Elisa, for that answer. So the next question is for Alyssa Galaro on ICH S12. The question is, what are the recommendations regarding how similar the product evaluated in BD studies should be to the intended clinical product?
Hi, thank you for that question. So it should be taken into consideration the manufacturing process, important product characteristics such as titer, and also the final clinical formulation. But one thing to note is that species-specific or orthologous transgenes can also be used if immunogenicity against the transgene is a concern, and that justification should be provided to the regulatory authorities.
Apologies. Thank you very much, Alyssa, for that response. Moving on to Chris and to Q5AR2. Chris, why have some gene therapy products, such as AAV vectors, been incorporated into the Q5A revision and not others, such as CAR therapies?
Well, thank you for that question. So the new scope applies to viruses that are amenable to virus clearance studies, and where the product is not impacted negatively by the virus clearance steps. So this immediately rules out cellular-based products such as CAR T-cell therapeutic products. However, non-envelope viruses such as AAV are resistant to some virus clearance steps, such as some chromatography-based methods, low pH conditions, and virus filtration. Envelope viruses, by contrast, are quite labile and sensitive to low pH conditions, detergents, where proteins within the envelope can become denatured or solubilized by these treatments.
Great. Thank you, Chris, for that response. The next question is for Larry Lee on Q13. So the question is, I understand that continuous manufacturing is a hot topic. I've also heard about advanced manufacturing. Are these phrases synonymous?
Yeah, thank you, Nick, for the questions. I want to just clarify that continuous manufacturing is just one example of advanced manufacturing, and advanced manufacturing can encompasses the novel method for manufacturing, but also advanced analytics, as well as novel dosage forms. Thank you.
Thanks very much, Larry. So back to Nick, and as the audience can probably tell, we're going on rounds for the entire group so that we can be sure to answer as many questions from as many folks as possible. So Nick, what's the difference between a guideline and a guidance, and where can I get trained about ICH guidances that have already been implemented?
Great, thanks for that question. So I'll take the first one first, which is fairly simple. So there is no difference between a guideline and a guidance. The two terms are definitely interchangeable. So in North America, we use the term guidance more, whereas internationally, the term guideline was used. So when the ICH organization was developed, there was an agreement that the international term guideline would be used rather than guidance, but the two terms are definitely interchangeable. In terms of where training can be provided, or where you can find training and where you can get trained on ICH guidance, there are a number of places. So I would first check with your regulatory agencies to see if there is training available. I know both at Health Canada and in US FDA do offer some training on ICH guidelines. Over the past recent years, ICH has been very focused on developing more global training on ICH guidelines as well. There has been on the ICH website, if you look under the training tab at the top of the homepage, you'll see that there is a new training library page that has been launched. That is very recently launched. Again, something that ICH is really focused on, and the ultimate goal is over the next 3 to 5 years for on that training library page for there to be some training modules that have been posted on that page on almost all ICH guidelines. So definitely have a look there as a first step. Thank you. So moving on, excuse me, to the next question is for Anna Edmison on M10. The question is, for stability evaluations, why is it necessary to measure the concentration of the QC at time 0 if the assessment is based on percentage nominal?
Thank you. This is a question that often comes up. An initial assessment is required simply to verify that the QCs were prepared properly before they're subjected to the stability assessments. Thank you.
Thanks very much for that. So the next question is for Vivian or for Ron on M11. It's great to see this protocol harmonization. Could you please speak to how this effort will be similar or different from the NIH FDA protocol template and the Trancelerate electronic common protocol template, and any efforts to collaborate on these efforts. And the questioner also added, thank you for this important work.
Sure. So we are certainly on the expert working group familiar with both the NIH template as well as the Trancelerate template and a few others. One of the first things we did was take a kind of a scan of the landscape to understand where there might be sources of material that might inform our design for an ICH protocol template. Those two were among the sources that we consulted, and I think it's fair to say that we were influenced by them, not exclusively by those two, but if you compare them, you can see that there's some influence happening. So it's not identical, but you know, I would say it would be fair to say we built on many of the concepts that you find in those two. We extended it further in terms of getting to a true technical specification. The Trancelerate template has some technical aspects to it, but we're going quite a bit further than they did, more completely and more thoroughly. So I think it's a big step forward, but you know, we certainly learned a lot from the work that those groups did.
Great, thanks very much, Vivian, for that response. So the next question is for Raj on M12. So the question is, there are some differences in cutoff criteria, in brackets static models, between FDA DDA guidance and ICH M12 draft guidance. What is the FDA's position until ICH M12 guidance is finalized?
Yeah, that's a very good question. As you know, that the draft guidance or guideline is primarily meant for seeking public comments and help resolving. And currently, the FDA final guidance is the one which we are implementing. Having said that, the ICH M12 draft guideline represents the most contemporary thinking. And given that we are always open to applying the most contemporary thinking, we would be reasonably amenable for utilizing the one which we are suggesting in the ICH M12 draft guideline at this point of time as something to consider.
Thanks a lot, Raj. Moving on to the next question, and this one I will pose to Lei. Lei, the recommendations for fasted fed studies in M13A appear to be in line with previous Health Canada EMA requirements, but not to those of the FDA, which recommends most times to do both fasted and fed, regardless of risk of product, if the drug is to be taken without regard to food. This would be a big change for FDA to drop the fed requirement for so many IR products. Do you know if OGD is really on board with this ICH guidance and would be making this change in the future?
Thank you, Joe. Thank you for this question. So as noted in this question, the recommendation in the M13A draft guideline with regard to the selection of the type of B studies, i.e. either fasting or fed or both, and the mu types is different from FDA's current guidance on BE. And this probably, as the person recognized, represent a key harmonization point through M13 work, because FDA is currently different from other regulatory agencies, including Health Canada and EMA. In the draft M13A guideline, the ICH M13EWG was able to come up to a consensus to propose a risk-based approach for determining whether BE should be conducted under fasting, fed, or both conditions. As you heard from John's presentation, high-risk product is a new concept delineated in the draft guideline, and currently this concept is not in any of the regional guidelines. So, and FDA is part of the M13EWG, and FDA supports this proposal. However, as you also heard from Raj, right, ICH M13A similarly is in a draft stage and currently is under public consultation. So it is not ready for adoption or implementation. Because it's in public consultation stage, we encourage stakeholders to submit comments to the FDA docket before April 3rd to ensure that FDA and ICH M13EWG will consider your comments on this draft guidance before the group begin to work on the final version of the guidance. And also you should submit your comments, relevant comments to other regulatory agencies. I believe we will continue to collect comments all the way through most likely May 2023. In addition, I also would like to make a plug-in for this audience that FDA will hold a SBIA webinar in early May to discussing ICH M13A. In the webinar, FDA will provide an overview of the guidance, explain the ICH EWG's current thinking, scientific thinking on the selected topics, and also provide clarification on the FDA's planning on the implementation of M13A for generic drug applications. And the registration will open in March. So with that, I would like to thank you for this question and stay tuned. Thank you.
Thanks very much, Lei. Moving on to some S1B questions and back to Elisa. Elisa, when the weight of evidence assessment indicates that a 2-year rat study is not warranted, do we need to provide human genetic association studies along with consideration for cancer gene databases? And is there any guidance on how this information is to be presented?
So thank you for this question. So there is no specific need to provide the results of human genetic association studies or information from cancer gene databases in the weight of evidence assessment. However, this information, if available, may contribute to the weight of evidence assessment. And there is no template or guidance on how this information should be presented, so sponsors can use any format they consider most appropriate and useful. Thank you.
Great, thank you, Elisa. So the next question is for Alyssa on S12. Question is there further guidance on the recommended number of animals for BD studies for gene therapy products?
Hey, thank you for that question. So as Sharon mentioned in her presentation, an appropriate number of animals should be evaluated at each time point to generate sufficient data to support the BD assessment. And it's noted in the guideline that this is a recommended of a minimum of 5 rodents or 3 non-rodents per sex per group per time point. But the guideline does mention that with keeping with the 3 Rs, the total number of animals can be aggregated from several studies. So justification should be provided for the number per time point and also the use of any data that are combined from multiple studies.
Thanks very much. Moving on to another Q5A R2 question for Chris. Chris, will sponsors be expected to perform a head-to-head comparison between NGS and the in vivo method when planning on using NGS for assessing virus safety for future submission?
Well, thank you for that question. It's an excellent question, and the answer is no. Sponsors will not be expected to perform head-to-head comparisons between next generation sequencing and the in vivo method. So all that will be required for submission is to submit a validation package that demonstrates that the use of NGS is fit for purpose. So the guidance revision encourages sponsors to have discussions a priori regarding the requirements for the validation. NGS, of course, as was discussed within the working group, that it can be regarded as a large improvement over the in vivo method, both in terms of the sensitivity and in light of the 3 Rs initiative. Thank you.
Great, thanks, Chris. The next question is for Larry on Q13. So what are you doing to promote the adoption of the continuous manufacturing technology?
Yeah, thank you, Nick. Thank you for excellent questions. We, from CEDAR perspective, we are trying to promote the adoption of a continuous manufacturing through three major efforts. The first one is we established the Emerging Technology Program, which gives an opportunity for the sponsor or manufacturer to engage early with us, so the FDA can provide recommendation or comments on the proposed new technology. And basically, this type of early engagement will help FDA as well as the industry to understand each other, to work together to bring the new technology forward. And then the second effort is to we are looking at to modernize our policy as well as the regulation to make sure that our existing regulation or regulatory framework will account for the advancement of technology. And then one of the initiatives we have is called the so-called FRAME initiative. And the Q13 is really definitely is part of the FRAME initiative to provide more regulatory expectation on how to develop and implement this type of a technology within the current regulatory framework, and also try to put some harmonized expectation in this area. So from the global harmonization perspective. And then the last one is we also working with academia and others, external stakeholders to conduct research to generate data and knowledge in order to help us to develop our policy as well as our assessment of the technology under the Emerging Technology Program. So that's the three major areas we are trying to focus on in order to promote the adoption of this type of new technologies. Thank you.
That's great, Larry. Thank you. Back to Nick. Nick, you outlined the process in which ICH selects new topics on an annual basis. What type of criteria are considered for ICH when selecting new topics for guideline development?
Yeah, that's a great question. So we do have a number of criteria that are considered by, as mentioned, a subcommittee of the ICH Management Committee. So there are things such as whether the topic is relevant from a global impact perspective. So is it relevant to regulated activity across all ICH regions? Does it have a clear case for reducing regulatory burden? And does it obviously to industry and regulators, which is important to have on both sides, but also is there a risk exposure to human subjects or patients? Is there an urgency or timeliness? I think this one's important as well. If the proposal really is related to an area of interest of increasing regulatory policy concern or public interest, are there any legal barriers? I think this is one important thing that we've noted across the board, especially from our regulatory perspective, that these topics really have to be compatible with the provision of current national laws and regulation across ICH regions. We aren't looking to impose regulatory change on any regulators. We know that is a very onerous process. So something that is feasible to implement across ICH regions. Practicality is also important as a proposal in alignment with active ICH projects and current ICH guidelines. And of course, the technical aspects, is there a sufficient scientific understanding whether the topic area is mature enough to be taken up, or whether the topic is still undergoing some level of experience gaining or knowledge gain. And from a logistical perspective as well, are the subject matter experts available to engage on this topic across ICH regions? So that's just a bit of a flavor of the discussions that we have within the ICH Management Committee when these topic proposals come up. So we'll move on to the next question, which is for Anna on ICH M10. So the question is, when will the ICH M10 training slides be published?
That's a very good question. We're working on this virtually, and while I can't give an exact date, I can say that we are currently doing slide edits, so stay tuned.
That was a very quick response and very thorough. Moving on to our next question, and it may be our last because I note that we are getting very close to our closing time. So I want to throw this, this is an M11 question, and I'll throw it to Vivian or Ron. The question is, to fully utilize the technical capabilities of the electronic protocol, will there be a specific software made available as an open source? In data exchange, how proprietary information can be protected. So with that, we'll turn it over to Vivian or Ron.
Hi, so an open source specific software solution is not one of our deliverables. We are delivering an non-proprietary data model and data standard, but we are not delivering a specific solution. Our intention is to equip software vendors or private companies to develop their own solutions. So we want to enable that kind of a future, but we certainly won't do anything to lock anyone into a particular technology or a solution, but to keep it open.
Thanks very much, Vivian. And with that, I want to turn it over to Ray in just a second, and to thank the audience very much for sticking with us on a Friday afternoon, and really appreciate your interest and your participation. So with that, Ray, I think you have some closing comments.
Thanks so much, Jill, and we want to thank all of our speakers for their very informative and timely talk, and also for responding to numerous questions that came in. Just a few closing reminders. This activity is eligible for an attendance certificate, which supports continuing education by SOCA, RAPS, SQA, and ACRP. Please refer to our website at fda.gov/CEDAR-SBIA for more details. You'll be able to obtain the attendance certificate only upon completion of a survey, which will remain open for 2 weeks. Remember, that's March the 10th. Please remember to download the certificate immediately upon completing the survey. It cannot be retrieved after the fact. Let us know your comments and feedback via the survey. This will help guide the design of our future events. Additional tutorials are available through fda.gov/CEDARLearn. On behalf of CEDAR Small Business and Industry Assistance, we hope that you enjoyed today's presentation and found it helpful. We look forward to your presence at future webinars, which will be advertised on our website at fda.gov/CEDAR-SBIA. Thank you for attending, and this concludes today's public meeting.